Good morning, everyone.
Good morning, Gerard. You can go ahead.
Okay, thank you. Good morning, everyone. My name is Gerard Salir. I'm the Director for Biodiversity and Coastal Research at the Department of Forestry, Fisheries and the Environment in South Africa. I want to say a very hearty welcome to everyone that's attending this workshop. Just looking at the attendance list, and there's a wide ranging of people coming from various countries. I'm thinking of Tanzania, Mozambique, South Africa, from the islands, the Comores, Mauritius, and then also as far afield as India, Pakistan, etc. I also see that there's quite a wide ranging of institutions involved in this workshop that's attending, including universities, research institutes, government, as well as NGOs and industry, with far a wide range of disciplines represented, from data managers to managers, researchers, students, research managers, which gives us quite a extensive stretch of people being involved in this workshop. I just want to thank everyone for attending. WIO for assisting with getting this workshop up and running, and all the different institutes that is involved with us. I want to say thank you very much. I think this is not only an opportunity to share information and learn a lot of new technical aspects, but this is also an opportunity for people to discuss different research projects and programs, and to get stimulated, to get people's research thought stimulated, going into the United Nations Decade of Oceans Science and Sustainable Development. So I want to thank everyone for attending. Next up, I'll introduce, it's Mr. Ashley Johnson. He's a colleague of mine. He's responsible for managing the Directorate Oceans Research. He's also the Vice Chairperson of the IOC of UNESCO, and is a member of SCORE and GOOSE. Basically, he was instrumental in getting the IIOE on the African continent established. And I want to say thank you very much for his attendance, and I'll hand over to him now. Thank you very much. Enjoy it.
Hi, and good morning to everyone, and welcome to the workshop. Thank you, Gerard. I think it's a pretty impressive collection of people, and I thank Tanya and her team, but also, of course, a special welcome to WIO, who's been so generous in their support of this particular workshop. Just a very short presentation from my side, and basically the title is about the second International Indian Ocean Expedition, which is really the reason why we got the data and the information that we have looking at present, and where to from here. Next slide, please. As we all know, we did the IIOE2, as it's commonly called, based on a regional priority, which we as South Africa didn't set, but we set it as a collective, as a region, and as different countries. We identified these national priorities, and this was all obviously under the subcommission of the IOC, and we produced this document. And the cruises then proceeded into the region, with many of you who probably are here today in attendance on those cruises. Next slide. During this process, I think it was important for Africa to leave what we would call an African legacy, which did not happen in the first International Indian Ocean Expedition in the 1960s. So we've explicitly gone out and set about to create these hubs, which we called centers of competence in the different countries, like Kenya for data, South Africa for operational oceanography, Zanzibar for biodiversity, Mauritius for remote sensing, and obviously still identifying an area in the west of Africa to do another area of competence, because we did not want to lose the opportunity this time around for Africa to make a significant input into ocean sciences, which I think is incredibly important. Next slide. So these were the cruises that we undertook in 2017. As you all know, there was quite an intense survey in Mozambique and into the Tanzanian area. Next slide. And then in 2017, we had a wonderful list of people from all over the African continent and even as far afield as France to join us in holding our collective hands as a team of African scientists doing African priority science within the ocean space. Next slide. 2018, we then did another cruise into the region, again, the Tanzanian, but this time focusing on the Comoros Island as well, because we know that Africa has a certain lack of operational oceanographic data, but very often our cruises that are done within the region are done by foreign vessels, but this decided that the African countries will dictate their own priorities and we will undertake the science we have decided as a collective as what is important for ourselves. Next slide. And again, we had a wonderful team of people from all over Africa. And of course, it was a large contingent, particularly from the Comoros, because there the opportunity was to do from the biodiversity benthic, which is what is worked out through water column dynamics, through water column biodiversity, as well as the oceanography and everything that was required within it. Next slide. So going forward, I think the workshops that we're doing at the moment is an important step for young African scientists, but also established African scientists to participate in an international program. But the important thing is it is based on the priority science that we have decided for ourselves. And I think that is an important point to take away. And what else is important is that we as South Africa, but also as the region and as a continent, see the International Indian Ocean Expedition as a stepping stone. It was the time for laying the foundation of African science and African priorities as a stepping stone into the United Nations decade of ocean science for sustainable development. And I would like to iterate the point that it is African priority, African science, so that we as a collective can make that scientific input into international programs, that we run our own destiny, that we develop our own people, so that we land up with an ocean economy, but also a conservation priority for Africa by Africans. And with that, I thank you, and I hope you have a wonderful time. And I hand you over to Dr. Tanya Haupt, who is instrumental in all of this, who's put it together. And she too was a participant on these cruises and playing an important coordinating role. So I wish you well and an amazing workshop and outcomes. And I thank you very much. Over to you, Tanya.
Good morning, everyone. Thank you, Sharon and Ashley. Thank you all for joining us. Welcome to the Western Indian Ocean. Regional Benthic Imagery workshop. In my talk, I'd like to brief how this workshop firstly stems from the IIOE2 expeditions and also provide some background as to how we find ourselves here today. We'll now switch to the presentation. In the years 2017 and 2018, the South African Polar and Research Festival, the SA Agulhas 2, undertook regional training surveys in the waters of Mozambique, Tanzania, and the Comoros. These expeditions, hosted by the DFFE, aim to capacitate young African scientists in various marine disciplines, including that of benthic biodiversity, all through the collection of data and information in the Western Indian Ocean. Participants that had joined the benthic biodiversity team were trained in a variety of standard and advanced research methods, from collecting, processing, and identifying physical specimens to using non-destructive benthic camera systems to obtain images and video footage of the seabed. From these IIOE2 training expeditions, we realized that even though marine researchers worldwide are using these tools in the exploration of benthic life, there was a need to capacitate African scientists within the Western Indian Ocean region in how to use an underwater imagery platform to better document benthic ecosystems. We saw a way forward through WIO's Marine and Coastal Science for Management program, also known as MASMO, and through a successful funding application obtained at the end of 2019, the Western Indian Ocean Regional Benthic Imagery Workshop came to be. Not only was this an opportunity for capacity development within the Western Indian Ocean, but it was also an opportunity for participants to get involved in processing and analyzing the data collected on the IIOE2 expeditions from their respective countries. Researchers, technicians, and students from the Western Indian Ocean could apply to attend the regional training workshop that was to be held in Cape Town in October 2020. Unfortunately, our plans were disrupted when the COVID-19 pandemic hit. But instead of taking a step back, we saw an opportunity to host this training event virtually and reach a far wider audience than what was previously possible. And as for the IIOE2 data, through a collaborative effort managed by the DFFE and the Ezekiel Museum of South Africa, we now work remotely with the principal investigators from Mozambique, Tanzania, and Comoros to process and analyze this data, the output being a specimen occurrence and image observation database. A brief outline of how the week will run. So by now, you should have all received a detailed program indicating the sessions for each day. The main objective of this workshop is to provide information or training on how to use benthic imagery for research. And besides raising awareness of the utility and importance of this methodology, we also touch on how to mitigate the limitations of the technique through using taxonomy and collections as a verification tool. We will start off with a short session in which we will have an introductory talk to provide a quick overview of benthic invertebrates and the preliminary findings of the IIOE2 expeditions. You will also get to learn more about the WIO Benth project, after which we will move on to sample design, and thereafter data collection, data processing and analyses, and lastly, data management. There's also a session dedicated to bruvs for those more interested in fish communities. And because we couldn't bring any of you to Cape Town for the actual practical training component, we have produced step-by-step training videos of some of our underwater camera systems. So do not miss out on this. The last day is dedicated to interactive discussion sessions. So where do we go from here, or what challenges are we faced with? Now, this will take place in a brand new venue, meaning that on Friday, you will not join this webinar, but rather we will all join in a new Zoom meeting. The link for this meeting is in your program and will also appear in the chat box throughout the week. Within this meeting room, we will have various breakout rooms, each focused on a particular topic. You can visit the room that you're interested in, or you can move between rooms if you wish. The topics range from a best practice manual for habitat classification in the main room, room 1, challenges with imagery data in room 2, to building collaborations and infrastructure in room 3. Room 4 will leave open as a general let's chat about things room. This all happens on Friday morning. It's a chance for all of us to network and to hear what your experiences and challenges are and discuss what the possible solutions could be. We will finish the session before lunch, after which we will close the workshop and communicate further with you via email. On that note, I'd like to use this opportunity to thank all those that have made this training event possible. Firstly, WIO for providing us with the funds to host this workshop. We've had to completely change the way we envisioned it would be, from our initial plan to bring candidates from all around the Western Indian Ocean to Cape Town to board a big red ship, the SA Colors, to a virtual event where we interact with you through our computer screens at home. Crazy times indeed, but throughout we had the continuous support and understanding from the WIO team. So thank you for this. Also to our managers and directors at the DFFE for supporting and allowing the Benthic team to host this event. A big thanks to our team of administrative ladies, to UWC for managing the funds, our technical support at SciFest Africa. Thank you for all your time and patience. You have opened up our eyes to this virtual world where the possibilities are endless. The film team at Octopi Africa and Array Media for producing the practical training videos, together with our colleagues at SciHab, Scion, and UKZN, the curation team at the Zika Museum of South Africa, our Benthic Biodiversity team at the DFFE for all your support, your input, and hard work whenever it was needed, and to all our colleagues, the presenters who have taken the time to prepare for this workshop and who will be sharing their expertise and experiences with us. And of course, to all of you for joining us. Thank you, everyone. Have fun. We encourage your active participation in hope of a rewarding training experience for all. I'll now hand over to Dr. Matthias Sigulu, who manages the Marine Science for Management or MASMA grant at WIO. Dr. Matthias holds a PhD in functional marine ecology and has been involved in various projects surrounding coastal and marine resource management in the Western Indian Ocean. Over the last 4 years, he has been leading the MASMA grant as the WIO Secretariat. MASMA is the regional flagship program that supports emerging and senior scientists to turn their dreams and ideas into practical solutions that contribute to the regional and international development agenda. Dr. Matthias and the rest of the WIO team, thank you for your support in making this regional training workshop possible.
Well, good morning, everyone. Wherever you are sitting, whether it's in the morning or in the afternoon, but I hope you are all doing well. We from WIOMSA, we are very excited to see that this meeting is finally happening. It has been a long waiting, but it has been long waiting for good. And I see today we have almost over 150 participants. If it could have been a physical meeting, it's not possible to have this number of participants, but given the circumstance, we are able to accommodate many people. So first of all, I would like to thank the Department of Environment in South Africa, the University of KwaZulu-Natal, and all other institutions that have participated in the building up of this particular workshop. Special thanks goes to you, Tanya. I know it has not been easy, but finally we are here today. And we are looking forward to successful implementation of this regional workshop. So from the WIOMSA side, the Executive Secretary, Dr. Chuda, was to attend this particular meeting, but he is also attending another meeting, but he could not make it here. But he is looking forward to the successful implementation of this particular workshop. So I don't want to spend so much time because I see we are still we are a bit late of time, but we hope we will catch up. But for us, this the first workshops that we are holding since the pandemic, and it's the proof that we can still work in the region, not physically, but visually it is possible because the conservation of our marine habitat till the pandemic is over. So the effort must continue despite the current circumstance. So we hope this will build up a big network for people working in Benthic subject, so to say. And this particular workshop is also linked to the MASMA projects that we are supporting on WIO Benth. So I see Shan is also here, is the PI of the other projects. So we hope this particular workshop and the final product from the WIO Benth will give us an image on what is there in the benthic habitat of the WIO region. So without further ado and not wasting so much of time, thank you, Tanya, and all people that has put this together. And we are looking forward to successful implementation of this particular workshop. So thank you very much.
Thank you, Mati. We'll now move on to some housekeeping rules. Presentation will play shortly. I'll now go through a few housekeeping rules or guidelines with you. So live translations will be available for most sessions from English to French. So even if you're English speaking, when you join the webinar, choose English, or if you're French, choose French. To do this, click the interpretation icon and select your language of choice. You can do that now if you haven't already. Today's webinar is being recorded. We will be able to share a link with you after the event where you can download the various sessions. We welcome you to revisit the content yourself and also feel free to share it. Also note that this event is currently being live streamed on WIO's Facebook page. Attendees' microphones will be on mute and videos will be off. This means that you will interact with us through the chat function or the Q&A box. The chat function is for technical questions or issues and loading of reminders and relevant links. So please do not place questions to the presenters in the chat box. Use the Q&A box to ask questions to the presenter, or alternatively, you may also raise your hand. And if there's time, you can ask your question verbally. Note that the raised hand option is only available to those tuned into the English audio channel. If you're on the French channel, please only post questions in the Q&A box, and the interpreters will translate this for you to ask the presenter. If you like a question asked for someone, or you have a similar question, then click the thumbs up icon. We will focus on these questions first, and questions that are not attended to will be retained to be answered at a later stage. In summary, be on time, ask questions, and encourage discussion on your question by clicking on the thumbs up. We also have a few poll questions, some fun, some informative, and a way for us to obtain information and also rate your experience of the webinar. So please participate in these. We will show you how the polls work shortly. If there's time, and if you're not seeing the polls, it means that you do not have the latest version of Zoom. We will post the links in the chat box for you to upgrade your current version. There are polls that have only one question and others that have multiple questions, which you will need to scroll down for to answer. So if there's time, we will demonstrate one or two polls in a bit, and we will start the first session of the day soon. So use this time to familiarize yourselves with all the functions and communicate with us in the chat box if you're experiencing any technical issues. Thank you, everyone. We will see you shortly. Thank you, everyone. That is just to demonstrate how the polls will work. So we will start shortly.
Good morning, everybody. A very warm welcome to all of you participating in the workshop today. My name is Dr. Alison Koch, and I am a marine biologist at the Cape Research Centre of South African National Parks, where I work mainly on marine protected areas. Just a reminder to please choose your audio channel, English or French, and then also to please use the Q&A function to ask any questions to the presenter. Okay, so let's get started. This session includes two introductory presentations on results from large-scale benthic surveys that have been conducted in the Western Indian Ocean region that we've already heard about this morning. The first presentation is by Robin Payne. Robin is a PhD candidate at the University of the Western Cape, as well as a research assistant and data coordinator at the Iziko South African Museum. She's currently involved in compiling a specimen occurrence database and species pages for South African sponges through the FBIP project, and is the research coordinator of the IIOE2 Invertebrate Specimens and Data Project. While based at DFFE as a student, Robin was involved in numerous sampling trips, including intertidal, SCUBA, ship-based collections, while also participating in assisting in marine invertebrate taxonomy workshops. Thank you.
Hi everybody, I'm Robin, and today this presentation is really going to focus on the preliminary findings regarding the marine benthic invertebrates collected during the WIO expeditions. As you can hear, I'm kind of losing my voice a bit, so please bear with me. And as we're still working on these specimens, I'll also kind of provide some observations and suggestions regarding the collection and initial processing of such samples carried out during fieldwork, especially at sea. So your marine benthic invertebrates are really organisms that live on the bottom of the sea or in the sediment, and they have no backbone. So they live either on the surface of bed forms, for example, rock, coral, or sediment, and these are the kind of known as epibenthos, or within sedimentary deposits, so your infauna. And they really comprise several different types of feeding groups, including filter feeders, predators, etc. And here are just some examples of such animals, including there's some sponges, there's some hydroids, a sea star, coral, a crab, a bryozoan, and a gastropod. So your marine benthic invertebrates can really be collected in a variety of ways, and this includes intertidal collections, beach cast specimens, as well as snorkeling and scuba. Scuba is usually, really carried down to about 30 meters. It's not really advisable to collect specimens that have been beach cast, as they're often desiccated and or fragmented, and their diagnostic features, so those features that are actually used to identify them, are often worn away. However, some early researchers actually use such specimens to describe new species, such as Rhabdomia spirophorus, and this genus name has changed. Now, this is a sponge species described in 1931 from the east coast of South Africa. So these kind of species actually need redescription with modern methods. I'll just put a picture of a knife here because it's always advisable to take a knife with when collecting, as many invertebrates actually contract and they may become very difficult to remove. So some ship-based methods include your sled, your grab, and your trawl, and this is for sediment or soft substrate, as well as your dredge for rocky or harder substrate. So most benthic invertebrates that we work on, or that I work on specifically, are really collected using the dredge, which can sample down to hundreds of meters, and which targets sessile animals that need something to attach and grow on, for example, rock. So this can include kind of animal groups like sponges or corals or things like that. So your dredge actually is dragged along like this to collect benthic invertebrate samples. So although the dredge can include specimens in from various kind of habitats, many of which actually become fragmented, it can also reduce bias, as you are not just collecting animals that are conspicuous. So for example, I find if you're doing intertidal collections or scuba, you might be drawn to the largest, orangest, beautiful sponge, where there might be some nondescript kind of encrusting, green sponge hiding under a rock that actually might be more scientifically interesting. So we really need such destructive sampling to obtain specimens for identification purposes. So for example, sponges actually show great morphological plasticity, which means that they actually change their morphology according to the environmental conditions that they're in. So they are actually very difficult, if not actually impossible, to identify from images or video alone. And this is especially true if the fauna of the region is actually not well known that you work in. Physical samples can also be further used for other research, such as genetics and or chemistry. And this really speaks as well to the importance of reference collections, as well as their management and conservation, which we'll discuss in a different presentation. Finally, I've just put some pictures of gloves here, because when you're collecting or sorting through your samples, always wear gloves. So some invertebrates actually have very interesting chemicals or features, such as these spicules found on sponges, and they can actually cause dermatitis. So for example, sponge spicules are actually made of silica, so you can actually end up with like glass splinters, which is definitely not fun. So somewhat less to non-destructive sampling equipment includes the drop or the jump camera, the remotely operated vehicle, and the ski monkey camera system. So I won't really go into detail, as such methodology or equipment will really be covered by some of the other presenters. but your images and your videos can really provide an idea of your species in situ. So you get to see the specimen or the species. It's not deflated, it's not fragmented, it might be its original color. And it also really documents possible symbioses and or habitat types. So just to go into some preliminary findings regarding the marine benthic invertebrates, there were 2 collaborative cruises that were carried out in 2017 and 2018. And these were really done on the SA Gallus 2, which is a South African ice-breaking solar supply and research vessel. So 40 stations yielded benthic invertebrates from a depth of about 30 to nearly 700 meters. Sampling was dominated by the use of the dredge. There were also some grabs and also a bongo. So this is an example of a bongo net. It's used in biological oceanography. So obviously I just caught some, kind of benthic invertebrates kind of by mistake that were included in our database, or in our data sets. Most sampling was carried out in Tanzania, followed by Mozambique, and then the Comores. So here's just a map of the stations which yielded benthic invertebrates. So station time and location was limited due to the multidisciplinary nature of the cruises. More specifically, the benthic biodiversity team were limited by extensive mud habitats, here demonstrated by a jump camera, ski monkey, and dredge sample images from off Tanzania. And these also kind of show riverborne detritus, so you can see some leaves and things like that that were obviously washed into the sea from a river system. On the other hand, sampling around the Comores was also limited by the rugged topography and steep drop-off. So for example, you're kind of dragging a dredge behind your ship like this, and if you have a rugged topography like this, if your dredge hits a pinnacle or an outcrop, it can actually just snap. Alternatively, your weak link will break, or your dredge will actually tip and all your sample will fall out. So sometimes you have to look at an ecosystem to kind of weigh up the risk, whether it's necessary to actually do the dredge in case you lose it. So once a grab sample is sieved, so basically your grab goes down and it picks up soft sediment like this and it brings it back to the surface. So you can actually sieve that sample with water and you can remove your benthic invertebrates. Or with your dredge, you just put the whole sample onto a top and then you can kind of remove your invertebrates like that and then you sort them. And then at the same time, you can capture your data. So for example, your station number, your depth, the date, that kind of thing. And then you would label your samples and photograph them and then preserve them in 96% ethanol. So it can be really useful to take these kind of group pictures as you get a visual overview of the station. So for example, this is one station, and these are all the kind of invertebrates from that station. It also documents color. So some of your invertebrates, as soon as you put them in ethanol, they lose all their color, and color is actually diagnostic for certain taxa, for example, hermit crabs. Guidebooks like these can actually be very helpful to identify specimens to higher taxonomic levels, such as your phyla. And this is also really useful for students and interns or participants which are unfamiliar with the groups. They might have come from a different scientific background, for example, biological oceanography. Now they're doing benthic biodiversity. So it can be a really great teaching opportunity. So I found this guide particularly useful. You might not be able to use it, as you can see, it's for South Africa. It's kind of identified to species level, but you can kind of get to phyla or higher family kind of levels. It's free online, but I'll try and make it available in the literature folder. But we also find it very helpful because it has these phyla overviews, which we printed out and put around the lab. And it also provides great tips with regards to collection and preservation for different taxa. For example, some of your groups actually need to be relaxed with menthol crystals before preservation. And so this is for future identification or future use of the specimen. And this can include groups such as joint enemies. So images and subsamples of the substrate from the dredge and the grab were also taken for classification and indication of habitat type. So we have been using the Katami classification scheme for consistency, with abbreviations made for easy entry into the field data sheets. So for example, your dredge might come up. It might consist of rocks, shell hash, and some gravel. And then you would just put all three of those abbreviations into your data sheet. And just for example, here's some rhodoliths, I think some from volcanic rock from the Comores. So today, the marine invertebrates collected during these cruises are actually housed at the Ezeco South African Museum, located in Cape Town. So here's just an image of the museum here. And this is actually where the majority of the specimens will remain indefinitely. So this is the kind of the workflow that we developed starting from April. And the invertebrates, or sorry, the specimens have undergone treatment. So they've been rebottled and relabeled, and they've been assigned catalog numbers. So these are unique museum numbers. The database has actually been consolidated from three or four different databases and has been reformatted according to Darwin standards, and it has been updated. So we have actually managed to sort our specimens to phylum level. So here's just an example image. So I think those are your arthropoda, those are your mollusks, and that's your peripheral, your sponges. And right now we kind of dealing with parataxonomy, sorting to morphotypes, and then sending images and or specimens away to the relevant taxonomists. So this kind of process, however, has been quite badly impacted by the pandemic because we have very restricted lab access. And just a note as well, our PIs, especially from the Western Indian Ocean, really are playing a vital role with regards to validating the database, as well as with species identifications. So overall, we actually managed to collect 1,095 sample lots. And we say sample lots because most jars include more than one specimen of the same species. So for example, here's a shrimp, so you would say maybe that's a specimen. Here, this jar actually contains maybe 2 or 3 sea stars of the same species from the same station. And you can maybe call that a specimen or sample lot. And as you can see, the majority of specimen lots were actually collected from Mozambique, followed by Tanzania and the Comores. So certain phyla are dominant, and this includes your mollusca and your porifera, so that's bivalves, gastropods, and sponges. Others were relatively well represented, and this includes your arthropoda, Cnidaria, and Echinodermata. So that's arthropoda is like your crabs, your shrimp. Cnidarians is like your coral, your hydroids. Echinodermata is like your sea urchins, sea stars. The remainder of the phyla actually had less than 40 sample lots each, and this includes the annelida, so that's like your polychaetes, your marine worms, your bryozoans, moss animals, chordata, which is your ascidians or your tunicates, Sipancula, which is your peanut worms, and then Brachiopoda, which is your brachiopods. So out of interest, here is just a plate indicating some of the specimens that were actually collected. For example, this sponge is assigned to the genus Phangophyllina, and it's quite actually recognizable by its circular form and its prominent oscule. So sponges are filter feeders, and the water actually has to exit somewhere, and that's where it exits, the oscule. And there are only 2 species recorded from the Indian Ocean. Another example here is a brachiopod. This could easily be mistaken for a mollusk, but it actually has unequal halves or valves, and it actually falls into a completely different phylum. Now there's some other examples. So here, there's a coral, sponge, there's a mantis shrimp. Your crabs are quite easily identifiable. You know, bryozoans, brittle star, and a really unusual little sea star. So we're really currently in the process of actually trying to get these specimens identified. However, the taxonomic impediment also plays a huge role. So we don't have enough researchers specializing in identification of different benthic groups, especially in the Western Indian Ocean region. But this is also a problem worldwide. Often another problem is that in reality, taxonomists are often not actually employed as taxonomists. So you can only usually do, identifications whenever you have some free time, which is obviously not ideal. And then even in South Africa, the situation is actually quite dire. So in the recent National Biodiversity Assessment, it was stated that the majority of taxonomic work is actually carried out by part-time or retired researchers and or postgraduate students. It was also suggested, quite sadly, that most of the remaining taxonomic expertise in South Africa is expected to be lost within the next 5 years. So that is just something to consider. So this is obviously very preliminary as well. The spatial variability of benthic invertebrate communities within the region was investigated by participants during the 2018 expedition, but they only focused on 12 stations from Tanzania. Analysis were run in primer using a presence-absent matrix, with invertebrate communities showing significant difference according to depth and substrate type. So these kind of analyses will be reperformed when all specimens have been identified to the lowest taxonomic level, so preferably species level. But other outputs from this kind of a project could include species lists and a specimen occurrence database. So in addition, specimens have been stored in 96% ethanol for potential future genetic investigation. And this may be especially important with regards to understanding genetic connectivity within the region, as well as to further resolve cosmopolitan species complexes. So this is the case for many of our benthic invertebrates, including things like bryozoans and sponges. We have species that have been recorded from with a kind of cosmopolitan distribution. So they're kind of recorded maybe worldwide or maybe from the North Atlantic and the Caribbean and South Africa. And it doesn't really make sense that those species with such a limited larval dispersal can actually be found with such a large distribution range. So those kind of species or specimens actually all need kind of genetic reinvestigation to determine or resolve what's actually happening there. So that's just a very brief overview of kind of what we're getting on with. But I would really like to thank DFFE for allowing me to participate in the 2018 WIO expedition, as well as the Benthic Biodiversity and Geology team, and they're kind of shown here. And they really made it the whole kind of experience memorable and enjoyable. And finally, I would like to just say kind of, watch the space. So as we continue to work with the specimens, we will likely discover new and unusual species, while also further strengthening collaboration within the Western Indian Ocean region. So thank you very much for listening.
Thank you so much, Robin, for that really interesting talk and an overview of what the expedition is starting to reveal. We've got several questions coming in. The first question is from Paris. Is it hopeless trying to ID sponges from imagery? And if not, what taxonomic level do you usually get to?
Sorry, I'm still losing my voice. So it's not entirely hopeless. The problem is if you have a very well-known region and you've had sponge taxonomists or researchers that have been working over several years in a region, they have a kind of good understanding of what sponge species are there, and then you can kind of identify from there. Unfortunately, in South Africa and the Western Indian Ocean, we don't really know much about the sponge fauna. So for example, almost every single time we collect, we're finding new species. And that's just even from the intertidal, scuba, everything like that, we're finding new species. So here I wouldn't really recommend getting to like lower levels. So for example, you can usually get to maybe order or family, and that's maybe better scientifically. But you can always do collections as well and then just confirm. So for example, for my PhD, we did extensive collections, and then we also did videos and surveys of the region, so we could try to connect the species that we've identified with the imagery. Yeah.
Great, thanks so much, Robin. We've also got a message for you from Fadile. Congratulations for all the good work so far. Is there any way to prepare? Is there any way forward to prepare field guide for the Western Indian Ocean resulting from this expedition?
So that would be amazing. It's all down obviously to time and resources and things like that. So what we're hoping to maybe do, once we have a species list, we might do like a preliminary guide. So what I've been thinking, I haven't really passed this on to the other collaborators and PIs yet, but once we have a species list, we can maybe ask them to put in a few diagnostic features for each kind of species. And then we of course have the images that we've taken in the lab and with some of the ski monkey and the ROVs and whatever. So hopefully we can link those two together. It probably won't be a very intense or detailed guide. So for example, one issue we have is with sponges. They often put into guides, photographic guides, but they don't include the sponge spicules, which is actually how you identify the sponges.
So we also need to kind of look into that, you know, how far or at what level do we really go, basically. Yeah.
Great. And then there's another question on databasing. Is there a database of taxonomists for marine benthos in the Western Indian Ocean? And because this seems to be a major challenge for many of the benthic ecologists in this region.
No, 100% agree. It's even like that in South Africa. It's everywhere. You know, we just don't have enough taxonomists working on these groups. So what I find very helpful, if you're looking at a specific group, I would look at worms. So a lot of sort of people that work on certain taxa have offshoots of worms. So for example, I use the World Peripheral Database. So that's sponge taxonomists from all over the world that curate and edit this database. So they put in papers that you can freely access, and all the contact details of the editors are there. And they also, especially that group, they actually post, they've got a list of every single person that kind of works on sponges worldwide. So you can actually track people down. But I think there's only one working in the Western Indian Ocean, a few in South Africa, so not many. Again, as well, I think there's a marine expert website which you can kind of go to. But yeah, you basically, what I find, you've got to what I do is I look for papers in the region. So, or even if not papers in the region, but researchers from overseas that have worked on species within that region. So for example, if you have someone in India who's worked on crabs from the Western Indian Ocean or the Indian Ocean, I usually just contact them directly because we really don't have a database or anything like that to contact taxonomists. And of course, a lot of taxonomists as well, it's time. It's really time. So you might contact someone, but you might be waiting a long time for your species identification. So that's another thing to consider.
It definitely sounds like a challenge, Robin. We've got another question coming in here. Could you recommend methods for sampling invertebrates related to seagrass habitats? There's an area of study in the southwest of Madagascar.
I would probably say it's pretty similar. So maybe your methodology I haven't really worked in seagrasses as a disclaimer, but your methodology would probably, you would change that. I don't know how you sample seagrasses, but if you collected invertebrates, it would be exactly the same. So I would recommend, obviously, you need to know your location, your date, the depth, if it's like intertidal, if it's under 1 meter of water, something like that. And then of course, once you remove your invertebrates, take images. You'll see that I'm doing another presentation in a couple of days on reference collection and management. So that will go more into how to image your specimens. Always best to store in the highest sort of ethanol percentage because you can always do genetics later. So pretty much the same principles will apply, but obviously for different taxonomic groups as well. That other guide that I posted is very helpful. So for, like I said, some examples, like your brittle stars, your anemones might actually need to be, they kind of need to be relaxed so you can work on them in the future. So they don't go all like, you know, crunched up in your bottle and you can't actually see what you're working with. So yeah, it'll be pretty much the same thing, collect, photograph, try to get to a certain level of ID, preserve properly, and then, but yeah, I would depend different on your different taxa, I would say, yeah.
Great, thanks, Robin. There's another question coming through here. For Nigeria, how many stations were covered in Mozambique and in South and North Mozambique? And did you cover soft corals and hard corals or only hard corals?
So I can't really get back to you now. I can maybe answer later with how many stations. I don't have that information with you right now. We did collect, there weren't that many soft corals, but we did get at least, I know, 2 or 3 specimens, maybe more. And then we also did get quite a lot more hard corals, but all different types. So I think we have a Selena coralline, if I'm not mistaken. We have some little cup corals, and yeah. So I could maybe try to come back to that question with regards to how many Nigerians from the Mozambique. Yeah, and of course we're also looking for collaborators. So obviously we want to get as much as we can out of the data, and so yeah, we do have someone working on the sponges and the bryozoans, not anyone working on the corals yet. So that would be get in contact with me.
Lots of work by the sounds of it. There's a question related to sea sponges. It's actually directed to the WIO community, but perhaps you know whether sea sponges are one of the funding priorities for this area of research.
Okay, I can talk to that. So actually, I work mainly on sea sponges. So the problem is they're one of those kind of unlovable groups. You know, it's not a charismatic coelacanth or, you know, that kind of thing. But you do actually get a lot of funding, especially with biodiscovery. So a lot of your invertebrates actually have very interesting chemical compounds, and that's especially true for sponges because they actually have microbes that live within their tissues as well. So a lot of our funding actually comes either from trying to document new species in the region, but then a big portion actually comes from chemistry. So if you get your samples, so what we do is we get our samples, we take sub-samples for taxonomy and for genetics, and then the rest we actually freeze and we send to the University of the Western Cape. And then there they do chemistry. So they try to find out if there's any weird chemical compounds that can be used for like future pharmaceutical applications and things like that. But unfortunately, funding with taxonomy, funding is always difficult. But yeah, so you always have to find a way to kind of make it, palatable to to the upper, and that's how we do that. It's mainly chemistry, yeah.
That sounds like excellent advice. There's another question here. Have you found any similarities between the samples found in the Western Indian Ocean with the eastern part of the Indian Ocean species?
Eastern. So if I'm understanding that correctly, so I've done a lot of work on South African sponges and then on some sponges from Walter Shoal, which is just south of Madagascar. It's a seamount. And we have found a lot of connectivity between Madagascar and South Africa, or like, so the Western Indian Ocean and South Africa. We're finding a lot of common species. The problem with that is the sponges have a very short dispersal or larval dispersal time in the water column. So it kind of seems a bit, so they're not really found in offshore currents and things like that. So it probably wouldn't be transported in eddies. So we're not really sure how they're getting down, or if it's maybe, in geological time and they've kind of speciated from that. So we really need to do genetic investigation there. Again, so from the eastern side, I wouldn't say, I know some invertebrate groups have connectivity with like Australia and New Zealand and things like that. We do have a few that are similar, a few species that we say are kind of from that side, but again, it's the current systems. So what's the chances of, a sponge larvae that spends 2 weeks in the water column getting from Australia all the way to South Africa? So we're not finding much connectivity there, but I think, especially in the future now, we're doing more genetic investigations to just see, how similar species are, if they really are the same species, or if they just, in the early stages of speciation from like Madagascar and South Africa.
Thank you, Robin. Thank you so much. That was really fascinating and a fantastic way to start off the workshop. It's now time for our second presentation by Dr. Sean Fennessy. Dr. Sean Fennessy is an assistant director and senior scientist based at the Oceanographic Research Institute and has undertaken South African East Coast marine research since 1989. He is a local survey leader on surveys by the Norwegian research vessel, the RV Dr. Fridtjof Nansen of Western Madagascar and the South African East Coast. He has extensive knowledge of soft sediment demersal fauna from the Western Indian Ocean region and works closely with scientists from Western Indian Ocean partner institutions. A very warm welcome to Sean. Thank you.
Good morning, everybody. My name is Sean Fennessy from the Oceanographic Research Institute on the east coast of South Africa. Thank you to my DFFE colleagues for the opportunity to present on Project WIO Benth, which I am leading. which involves the identification, characterization, and vulnerability assessment of benthic ecosystems in the Western Indian Ocean. You can see there are several other WIO institutions who are involved in this. We also have funding support from the Western Indian Ocean Marine Science Association. We have strong support from the EAF Nansen program. And we also have colleagues from the Institute for Marine Research in Bergen. So what is the background here? Well, marine habitat mapping focuses on creating maps of important physical habitat attributes and associated communities and identifying critical areas of conservation importance. And traditionally in this region, those involve coral reefs, mangroves, and seagrasses. And there's a plot of these that you can see for parts of the Western Indian Ocean. Yeah, these are the sort of traditionally recognized as critical habitats. However, by far the biggest area of the Western Indian Ocean shelf and slope is really unknown. Generally, mainly comprises unconsolidated or soft sediments, very large area, particularly where the shelf is particularly wide. And people tend to just look at these as one homogeneous habitat, but soft sediments vary considerably in the type and distribution. So if you look at some work by a colleague of mine, Fiona Mackay and Andy Green up at UKZN, this is a plot of sediment distribution grain size on the east coast of South Africa. And you can see that the grain size varies enormously over a fairly small area. And even the mud content can vary considerably over a fairly small area. And similarly, the communities that are associated with these habitats also vary. So again, this is some work by Fiona Mackay looking at the macrobenthic infauna in the Richards Bay area, about 100 kilometers to the north of Durban. There's some faunal communities off Durban themselves and off the Tugela area. And you can see that the composition of the fauna vary remarkably. So that's the infauna. And similarly, for the demersal fauna, which are the larger trawled fauna in the region. So some work done some years ago based on the activities of the Dr. Fridtjof Nansen vessel. You can see there's a considerable difference between the shelf and slope fauna. These are not only fishes, these are invertebrates as well. And there's considerable variation along the length of the coastline as well, from the northern Mozambique area down to the central region compared to the south. There are considerable differences. So these communities associated with these habitats can vary considerably. Now, South Africa has been progressing quite well with characterizing its benthic biotopes and assessing their vulnerability. So I'm sure you'll see this map later on, produced by Kerry Sink and her colleagues for the National Biodiversity Assessment. So you can see within the EEZ of South Africa, there are marked differences in the benthic communities and habitats, and they vary considerably. And the more you zoom in, the more apparent these differences will become clear. In the wider Western Indian Ocean, this knowledge is lacking. And this lack of knowledge about vulnerable offshore habitats is hampering marine spatial planning. And traditionally, offshore Western Indian Ocean habitats also receive the least amount of protection. What do we mean by offshore? Well, generally beyond the surf zone and out towards the upper slope. Those are considered offshore habitats, at least for the purposes of this project. And as a consequence of this lack of knowledge, decision makers are not really equipped to assess trade-offs between utilization of non-renewable and renewable resources and trading them off against biodiversity, conservation, and other extractive uses. So what are the research questions that we are posing? So we're asking, what are the seabed types along the shelf and upper slope of the Western Indian Ocean? What benthic communities are associated with these areas? What habitat features characterize these areas? And are there parts of these areas that are vulnerable to anthropogenic impacts? So what are we doing? Well, the project started in September 2019 and is due to end late in 2022. There's no sampling involved. We're basically analyzing existing data and interpreting it. And it's survey ship vessel data, survey data, particularly from the Norwegian Dr. Fridtjof Nansen. The focus is on the western part of the Western Indian Ocean, mainly because there's been very little benthic surveying elsewhere, and in particular bottom trawls. So these are the 5 countries that are involved. And there is the focus, as I said, on the shelf and the upper slope areas, also because we've got limited surveys that have taken place in deeper areas. And generally we've incorporated or included surveys that include acoustic scanning of the seabed, trawls, grabs, and CTDs. Localities that have had more extensive or intensive surveying, smaller localities, might form the basis of some higher focused projects within the project. So what are the objectives? They are to characterize and determine the extent of seabed habitat types of the continental shelf and upper slope in the western part of the Western Indian Ocean, characterize and determine the spatial extent of the benthic communities of these areas, create the first benthic biotope descriptions for the continental shelf and upper slope of the Western Indian Ocean, using this info on seabed habitat types and communities, and identify the location and extent of potentially vulnerable offshore marine habitats in the Western Indian Ocean. And the particular activities that we've been undertaking so far, well, obviously a lot of literature and data reviewing, particularly on biogeographic provinces, habitats, benthic communities, vulnerable and critical habitats, and putting these into context with other oceanic regions, beg your pardon. And this information has been housed in a Google Drive repository. We've also been establishing the availability, suitability, and spatial distribution of acoustic, CTD, grab, and demersal trawl data. And we need to access, we have been accessing those survey data from the Nansen data repository at IMR in Bergen, as well as from other surveys. So if we just look at the Nansen survey data from 2007 to 2018, you can see there's obviously been a considerable amount of acoustic scanning of the seabed using sophisticated echo sounders. A large number of CTD casts have been undertaken, and we have access to those data. Bottom trawls from the Nansen itself in the region of 500 that we have catch information for, and we've cleaned that data. And then grabs for fewer, and we're in the process of accessing some of those grab data. And the idea is to validate, clean the data, consolidate it into a regional relational database as a start. So if you look visually at the surveys, the Nansen surveys, particularly over this period that I've been mentioning, so the bottom trawls, fairly widely distributed, a stronger concentration in Mozambique, as you can see, less so in Western Madagascar, and none in Kenya, because the vessel hasn't traveled there for many years due to piracy concerns. The grab information, a bit more sparsely scattered around, very little in Western Madagascar, a little bit up in the northwest, and none for Tanzania and Kenya, a little bit in Mozambique and off South Africa. CTD data from the Nansen, a little bit more substantial and fairly widely scattered throughout the region. We've been looking at other data sources as well to look at the suitability of including them. This is some survey or range of surveys, in fact, done by the first Indian Ocean expedition way back in the 50s. And there's a real mishmash of sampling gear types and spatial distribution that we've examined to see if they are worth including. This is some Natal Museum benthic dredge surveys that were done in the 1970s and 80s. We've had a look at that as well. This is work that DFFE colleagues have been doing up in Mozambique and Tanzania in 2017 and 2018 on the Agulhas 2. And this region here is off Durban, on the east coast of South Africa, looking at some observer data from the commercial crustacean trawl fishery there. So we've got some quite nice information from those trawls as well, from that area. So let's have a look at the various data types that we've had access to, looking at seabed characteristics. In other words, the acoustic component of things. We've got high resolution data from the vessel itself, the Nansen, and processing of those data commenced in July 2020. A lot of work being undertaken there. It's volumes and volumes of data, a lot of work to clean it and validate it and so forth, and that's underway. Kenya, I mentioned, there has been no visit by the vessel, Nansen vessel, there since the 1980s, but they do have their own research vessel, the Mtafiti, and she has been undertaking acoustic surveys, and those data have largely been processed, only single beam data, not multi-beam, but still, as you'll see shortly, quite reasonable. We've also got some some low resolution data in the form of electronic chart data that have been digitized. Obviously, we will work with JEBCO as a base layer. If necessary, we can consider including satellite data depending on water clarity and so forth. The SRTM data is a possibility as well. Inferring the nature of the seabed from benthic biota, I'll talk a little bit about shortly. So in other words, looking at the trawl fauna and what sort of habitat they prefer living in. You can infer what the seabed habitat is like from that. And then we also have sediment samples from the grabs and so forth, particularly from Kenya, from KZN, and from Western Madagascar, and others we're in the process of obtaining. This is a plot of the tracks of the Nansen vessel undertaking its regular transects up and down the coast. And the vessel has its echosonders running while it's undertaking those transects. So those are some of the data that we included or we looked at. In some areas, this is a close-in of the east coast of South Africa. You can see some additional scanning or high-resolution multi-beam and backscatter scanning has taken place to interrogate the nature of the seabed. The digitizing of paper charts and so forth I referred to, so that's quite painstaking work. So each of these bathymetry points with their attributes, if they're available, have been scanned or in the process of being scanned, and they've been used to develop these sorts of plots. And that's been done on a fairly extensive spatial basis throughout the region. This is the work that the Kenyans have been doing based on their single beam surveys on their vessel. And as you can see, although it's single beam data, you can produce quite a reasonable representation of the seabed. So in some ways, they are a little bit more advanced as a contribution towards this project. If we look at the water column, basically the CTD sampling, the physicochemical characteristics, we've got the Nansen data as well as some other CTD survey data available. And we've also got some candidates, participants in the WIO Bent project who have been nominated to join a planned EAF Nansen course on processing CTD data, hopefully later this year. This is a plot of all the localities in our spatial area of interest in the Western Indian Ocean, showing where we have sampling stations through CTDs were cast, and you can see certain areas have got better coverage than others, but hopefully they will be adequate to produce a broad-scale climatology of the WIO region to help describe or improve the description of seabed habitats, including things like salinity and conductivity, habitability as well. If we look at the benthos, the animals themselves, there has been some grabbing, as I indicated, as well as trawl cylinders, which are basically stainless steel tubes attached to the foot rope of the trawl, which do contain some faunal information. These are a bit limited, though, as you saw from the plot of the benthic stations, the grab stations, particularly well represented in KZN, less so in Mozambique and Madagascar, and very little in Kenya and Tanzania. The trawl data is probably the most substantial as far as the fauna are concerned. And again, a lot of Nansen trawls are referred to, but also some other survey data from Mozambique, Kenya, Tanzania, and KZN. This is what it looks like. I think this needs updating, but still you can get an idea of the distribution of benthic trawl information that we have. To describe a little bit what I meant by benthic biota inferred seabed habitats, we've looked at Nansen trawl data from 2007 to 2018, which incorporated South Africa, Mozambique, Madagascar, and Tanzania. From Kenya and Tanzania, we have trawls from the Southwest Indian Ocean Fisheries Project as well that we can draw on, and that's given us in the region of about 850 trawls. Out of those, we have approximately 1,600 different taxonomic species or taxa, and those names have been validated, and included with them in the database are habitat preferences for each taxon, as well as elements of its life history, which could be informative. And out of that, looking at the preferences, the habitat preferences of each of these fauna, whether they prefer soft, mixed, or hard substrata, we can infer, based on the relative proportion of each of these preferences in a particular trawl station, what the habitat is like. And this is some work that my colleague Bernadine Everett has done, interpolating from the benthic trawl data to infer what the seabed habitat is like in the areas that have been trawled. So this is still preliminary at this point. And as the trawl information is increased, obviously we will add to it. Looking at the species composition data in a little bit more detail, these multivariate plots are of taxa per trawl, presence and absence information. And in this top left-hand one, we've got depth included as a driver. And you can see quite a nice clear pattern of increasing depth from right to left. This is of the 850 odd trawls for the region, so quite a strong indication that depth is a driver there. If you look at this top right-hand one, that's just aggregated a little bit by shelf, upper continental slope, lower continental slope, and so forth. So it's emphasizing that distinction between the depth categories. Less clear if you look at the proportion of taxa that prefer soft habitats, or degrees of preference, if you like. So the picture is less clear there. So obviously depth, is a major driver, but there are still some underlying patterns that we will tease apart. So ultimately, this is next phase, probably at some stage next year, we will get around to this. So the idea is to use GIS layers of the species, the taxa, the taxonomic distribution, and the seabed habitats to create biotope maps, combining the habitats and the communities, and then use species distribution modeling layers to predict biotopes for the areas that have not been surveyed. And together this will enable us to detect the location and extent of benthic biotopes for the WIO. And ultimately, we can identify vulnerable marine habitats, or preliminarily so. What outcomes do we anticipate? Excuse me. So improved marine spatial planning in the Western Indian Ocean, as well as improved capacity of regional scientists in processing the types of information that we're working with. We also anticipate increased or improved knowledge of the habitats and communities, which will facilitate management decisions, which will enable decision makers to decide on spatial trade-offs between extraction and conservation. And obviously this has got implications for blue economy initiatives, as well as assisting WIO countries towards attaining their Sustainable Development Goals. And the findings will also integrate well with other current regional scale initiatives, which I'll show you shortly. And this inventorizing analysis and interpretation of acoustic and benthic community data will identify data gaps to enable or to assist with future survey planning in the WIO. So what are the conclusions and lessons learned to date? There are large amounts of existing data available for this region, and they've often been collected at great expense, particularly if they are ship-based, from ship-based surveys. But many of these data are not well inventorized, organized, cleaned, and validated, and it's been a massive job to do what we've done so far. And often accessing these data have been quite problematic. Our next steps, we obviously will continue this process of inventorizing and cleaning and validating available data. We have been a bit crippled because of the pandemic, being unable to hold data analysis workshops, and we need to seriously contemplate those very soon, because we're more than halfway through the project, and we haven't been able to have many of these workshops at all. There are some EAF Nansen supported CTD analysis and multivariate analysis workshops planned for later this year as well. So some of the other regional initiatives I was talking about, obviously the EAF Nansen Science Plan, a lot of their themes we selected quite carefully when we proposed this project. Obviously the Sustainable Development Goals, particularly conservation and use of the ocean, are the obvious ones for this, for sustainable development. UNEP's Nairobi Convention provides decision support services for management, as well as for protection of coastal marine environments, and is also heavily involved with marine spatial planning initiatives in the region. Similarly, their inputs towards the Convention on Biological Diversity and the World Conservation Monitoring Center, particularly regarding ecologically or biologically significant areas, or the IPSAs, and obviously these feed into area-based planning as well. UNESCO's Intergovernmental Oceanographic Commission, obviously the data that we are cleaning and validating will be made available through these, the ODIN Africa and the OBIS systems. So they're a resource to be used by the region. The second International Indian Ocean Expedition, DFFE, is quite involved, certainly with the surveys they've been undertaking in the region, and this particularly forms part of their theme 6, the unique geological, physical, biogeochemical, and ecological features theme. Thank you very much for your attention, and I can take some questions, I'm sure, if the chair will allow.
Thank you very much, Sean. Lots and lots of hard work. We've got a question about the trawl data. Is trawl data dependable in the determination of the habitat or specific locality of benthic biotypes? For example, can they tell us where the biotypes exist and communities in the seabed?
Yes, good morning. Look, trawling is just one tool. The more tools you use to try and examine a particular habitat, the better or more confident you'll be in determining exactly what is going on in that particular patch of seabed. So it gives you a perspective, and certainly grabbing would give another perspective. Acoustic scanning would give you another, an ROV would give you another. So yeah, the more gear you put down, the more confident you can be in what you're seeing or what that community involves.
Great, thank you. Sean, you mentioned that there's been a lot of challenges with the data, access to data, large amounts of data, obviously collected at a large cost. Have you got any recommendations in how to address some of these challenges? And can you share that with us?
Sure. Yeah, we could probably spend a week on those sorts of things. Look, I think keeping records and curating those records and backing them up and standardizing the format in which the data are kept would be ideal. Doing that at a regional scale, very difficult. Every institution, in fact, the lower levels of the institution have their own priorities and ways, and they're often reluctant to change or to adopt another system, but that would be the ideal. Taxonomy, yeah, is a major problem, as we've already heard. So the more sure you are of a species appearing in your gear, the better. And we have found that there's been a lot of inconsistency with that, misidentifications. Even species that don't even occur in the Western Indian Ocean have been identified as occurring in a particular trawl or whatever. So yeah, standardizing and attention to detail and keeping up with taxonomy is critical, and marine scientists should be doing that, certainly those who work with the fauna.
Thanks very much. And Sean, I'm sure you can, I mean, with all the data that's being collected and all these different sampling around such a huge stretch of coastline, there's probably a lot of species that's been collected that hasn't been identified yet. Is there any way to know what sort of percentages we're looking at an identified species?
Yeah, I think Charlie Griffith's review was probably the last time it was done on at that sort of scale. So it was done several years ago, but it could well be redone and updated. You know, I'm not saying our project could do that, but we could give some indication of that. The fish is obviously probably the best known. But yeah, there are many different phyla that are completely lack of data for many of them. I think Robin's starting to make some progress with sponges, like Fiona Mackay with the Macrobenthic infauna. But I mean, you know, she's really confined to the east coast of South Africa. There's a huge area to cover. So, but definitely could revisit Charlie's work of some years ago.
Thanks, Sean. And in terms of that stretch of coastline, what do you think are the major threats facing the benthos across that region? Yeah,
there's not a lot of benthic fishing going on. Yes, there's some crustacean trawling in the region, but it's at a relatively small scale compared to what's going on the west coast of Africa. But obviously fishing traditionally is a threat, so there is that element. Oil and gas, increasingly so in the region, and particularly in northern Mozambique, southern Tanzania. There are oil fields. Some of those are very deep beyond certainly the depth that our project's able to work. So they are an issue, and also other non-renewable resource interests. Again, these are mainly in very deep water, manganese nodules and so forth. But yes, there's increasing interest in those from a variety of Indian Ocean countries. So those are probably traditionally the main threats. Climate change, obviously, not as easily measured, but increasingly a concern for the region.
Thanks, Sean. There's a question coming in about how can an up-and-coming scientist in the region gain taxonomic expertise for the identification of species in the Western Indian Ocean?
You need to find a passionate taxonomist, I think, and be mentored by them. And yeah, there are increasingly fewer of those around, unfortunately. If you've got resources to be able to go international and be mentored internationally, you know, great. If you can afford that. There are some local experts, at least in the region. South Africa has several. And it's a case of identifying those, find a group that you're passionate or really interested in. And it's often your internal passions that will drive you and that will promote your increased experience.
Thanks, Sean. There's another question coming in. You said there was a lot of data that have been collected by ship, but were not validated and cleaned. If possible, can you request funds from either WIO or anywhere to recruit marine scientists or students to work on those data, probably to add some value to that region?
It's part of what we've done with some of the data sets that we've looked at. So certainly the demersal fauna that occur in trawls have been pretty well cleaned and validated, mainly by my colleague Bernadine. So for the trawled fauna, I think they're not too bad. Not always taken down to species level. Sometimes we've stopped at genus because of taxonomic uncertainties. But for some of the other groups and samples, for example, the macrobenthic fauna, so reasonably well known for the East Coast, but Fiona is discovering new taxa all the time, massive numbers of them. So it is a daunting task. As I mentioned, Robin is making some progress with sponges, but again, that's probably confined to the East Coast and obviously the South Coast of South Africa. But yeah, the rest of the region, difficult. The samples are not always kept. Unfortunately, there's a difficulty in curating those samples from research surveys. So they're not always preserved and taken back to the labs for curation. So that would be a good start. Start to collect animals. Don't just think, looks like that, therefore it is this. Put it aside and freeze it, preserve it, and then work on it back in the lab.
Great, thank you. And Sean, you must have seen such strange creatures and such interesting things coming out of these surveys. Can you give us an example of one of the most interesting or fascinating discoveries on one of these cruises?
I haven't been on that many. I've been on the Nansen a couple of times. I think Western Madagascar, a trawl came up from quite deep water, I think about 600 meters or so. And it was just full of, obviously like stalked glass sponges. I mean, it was just, the trawl obviously made a bit of a mess of the specimens, but I could imagine that field down at 600 meters must have been spectacular. So if we had a camera able to get down to that depth, it would have been really special. I mean, some fishes are really incredible looking, tadpole fishes or jelly noses. I mean, they're really weird and wonderful looking animals. And again, it's best looking at them live if you can with a camera. So when you see them in a trawl net, they're certainly not the same. In fact, that applies to just about anything that's caught in the trawl. They do get damaged, so you don't really get an appreciation of how spectacular those animals are in their natural habitat.
Fascinating. I always look at those creatures that we don't get to see very often as weird and wonderful. So Sean, those are the questions that we have for you. And what we're going to do now is launch a poll just before the tea break. And it's a multiple choice. So please scroll down to answer the question. The poll will be online in a few minutes, in a few seconds rather. So just wait for the poll to come up, and then please place your answer to that poll. Okay, so there's the poll. You should be able to see the poll on your screen now. It's multiple choice. Please select your answers, and then also please scroll down to see all 4 questions. And it would be great if you could complete that poll now. The first question is, what's your level of understanding of the topic? Are you completely green? You have some basic knowledge, you have a solid background, or you're an expert? Question 2 is, why did you decide to join this event? Was it to build knowledge? Was it for networking opportunities? Did your boss or your supervisor make you do it? Any other reason? The third question is around, where in your career are you? Are you an undergrad student, a postgraduate student? Are you employed, or do none of those apply? And then the last question is, how old are you? And there's different categories there. So if you could please go ahead and complete that poll. Sean, if you are still online, there's one more question that has been asked in the box. If you would be able to answer that for us.
Yeah, I was just typing an answer, but I can answer it live. I'm not aware of anyone who's looking at ascites in particular. They do form a substantial part of beach seine catches, particularly in Mozambique, excuse me, and I suspect then also Kenya and Tanzania, although in some areas beach seines are not allowed. But they are an important part of beach seine catches. Nobody's looking at them specifically that I'm aware of, but you could perhaps get some idea of trends in those catches if you're a bit concerned about their disappearance. Just need to be aware that sampling gear or fishing gear, I beg your pardon, might have changed, mesh sizes or whatever, so you might get distorted trends or patterns in the occurrence of the CTs in catches. So just be aware of that. But yeah, they're an important component of catches, small scale and subsistence catches. So yeah, there's probably data around to examine what you're concerned about.
Great, thanks so much, Sean. We've got one minute before tea time. We're going to be taking a tea break up until 11 o'clock. You should have been able to see some of those results from the poll. They've disappeared from my screen for now, but we are running on schedule, and I really want to say thank you so much again to Sean and to Robin for such fascinating talks, and for really starting off the workshop with that sort of large-scale picture of what benthic imagery can do, and the kinds of challenges we might be facing. So thank you very much to the presenters. Thank you again to WIO. And we're now going to have a 20-minute tea break, and we'll see you back here in the meeting room at 11 o'clock. Thank you very much.
Right, hello everyone. My name is Charles van der Meyden. I'm a lecturer and benthic ecologist at the University of KwaZulu-Natal here in Durban. And of course, welcome back. I hope you've all had been able to grab a cup of coffee or something. Before we just get back to things here, can I just remind everyone to, to choose your language in that panel, and obviously also if we can just keep the Q&A box for questions and the chat for more technical queries or problems. So this session between now and lunch is going to focus on survey and sampling design, and as well, also marine protected area design. And so first up, we have Dr. Denham Parker, who is a fisheries stock assessment scientist at the Department of Forestry, Fisheries and Environment here in South Africa, in Cape Town. And his mandate is to provide fisheries management advice to ensure sustainable exploitation of fish stocks. But his interests are broad and include marine ecology. And within that, researching population dynamics, the stock assessment aspect, and overall effects of marine resource use is his primary focus. He's currently the vice chair of the IOTC Scientific Committee, the chair of the IOTC Working Party on Billfishes, chair of ICAT South Atlantic Swordfish Group, and chair of the Lionfish Scientific Working Group. So welcome, Denham, and let's hear from you.
Good morning, everyone. My name is Denham Parker. I am a fisheries scientist at the Department of Forestry, Fisheries and the Environment, as well as a research associate at UCT. And today I will be giving you a very brief introduction to survey and sampling design in marine ecology. This talk is really an introduction. It's not exhaustive by any means, and I suggest that before you go ahead and develop any sort of sampling design, you do some reading of your own. But there are a few basics I will be touching on, which include defining a sampling unit, the different types of sampling designs, trying to estimate or figure out how many samples you do need or what your sample size should be, as well as a common mistake that's overlooked in sampling design, which is spatial autocorrelation. So yeah, let's get started. Study design refers to the number and distribution of suitably defined sampling units. Sampling units is something we will define later, and/or observations to be made on each unit for the purpose of answering a specific research question. So you have your research question and you set out to collect information best suited to answer that question. There's two aspects you need to consider. It's the spatial distribution and the temporal distribution. So you want to make sure that the observations you collect are spatially distributed across your population, and they are also temporally distributed across the time frame which you expect to see ecological changes in your population. The overall goal of study design is to provide the most efficient and precise estimates of parameters of the population that you are studying, given the practical and logistical constraints of sampling in the marine environment. So imagine you wanted to know how many fish were in a given area. Well, the best way to do that would be to count them all, but that's not logistically possible. Fish move, they are underwater. It simply can't happen. So you subsample. Or how much do you subsample? That is largely up to the logistical constraints. So really the goal is balancing a balancing act between wanting precise estimates of parameters, but acknowledging that we are unable to have unlimited funds and time to sample. So we're constantly trying to balance these out. And really, when we talk about the precise estimates of parameters, what we are saying is we're trying to understand variability amongst observations. There's multiple ways of doing this. In experiments, it's quite easy. You eliminate the source of variability or you fix the source of variability. Unfortunately, most of our ecological research studies are not experimental in nature, they're observational. So what we have to do is model the source of variability after collecting our observations. So the study design is really meant to make sure that we have the best opportunity to model the source of variability after we've collected our observations. So as a very basic definition, sampling is a process of gaining inference about a chosen population. Therefore, sampling design is the process in which we carry out prior to sampling to ensure that the data that we have at the end of the sampling process has statistical rigor. And what I mean by that is that you design your sampling in such a way that you do not violate any sort of statistical assumptions like independence, for example. And in this way, at the end of collecting all your observations, you have some level of confidence that your data is going to give you inference about the population that is accurate and precise. Therefore, clearly defining the population that we want to sample is critical to ensuring that we have sufficient and representative samples within that population. And really, the population needs to be described in terms of three internal attributes: spatial extent, temporal extent, and ecological extent. And I will go through each of these in a little bit more depth. Every ecological study has a spatial dimension to the research question. We generally refer to this as the study area. So the study area is the area in which you demarcate and you will collect observations from within. We should also always clearly define the spatial extent from which our findings are to apply. So collecting information within your study area doesn't mean that the inference from that will hold true elsewhere. But really, the important implication of spatial extent is, in sampling design, is selecting sampling units, both the number and the size. In general, your sampling unit must be compatible with your study area. So the larger the study area, the greater the number of samples needed to adequately represent the variation across the area, since larger areas typically are more variable. That said, compatibility is an issue. You cannot have a large study area with a small sampling unit, because it means you will need an excessive amount of sampling units to get representation, which is not logistically feasible. So again, the important part here is that your sampling unit must be compatible with your study area in terms of logistics, as well as in terms of being able to be representative. And I have an example there of, it is catches of tuna within the large pelagic longline fishery. And the top graph is within South Africa. You can see this, the South African waters, which is our study area, is clearly defined, and we see some definitive patterns. However, when we include that data into global catches of tuna within pelagic longline fisheries, it really doesn't even make a dent. You can see the majority of fishing is happening, or the majority of catches is happening in the tropical areas, particularly within the Atlantic Ocean. So it's just a very basic example of how, depending on your spatial extent and the resolution of the information that you gathered, you can get two very, very different ideas of what's happening. Similar to space, every ecological research question has a definitive temporal extent, how long you expect, or the time frame in which you expect to sample observations. If the question involves a dynamic that occurs over a decade, for example, then it's likely that the sampling will need to take place over 10 years or a longer period for you to be able to fully observe the extent of that dynamic. For example, we cannot examine the effects of climate cycles on marine invertebrates by sampling climate and invertebrates in a single year, because the dynamic simply takes decades. And this is a reason why we have such trouble understanding climate change effects. So the figure on the right top is the sea surface temperature for a year in Tsitsikamma marine protected areas. And it's a good example as to why most ecological studies take place over a year or longer. The reason being is that most environments have seasonal effects to them. And for you to fully understand them, you need to observe the implication of these seasonal effects on the environment and your study sample or your study area. The third and final aspect of describing your sampling population is the ecological extent. What aspect of the ecological process within your sampling population do you wish to sample? It can be very simple, such as species-specific abundance data, in which case you literally count the number of species or the number of individuals of a particular species that you observe. You can look at specific ecological processes, such as a predator-prey relationship, in which case you need to definitely get abundance data on both the predator and the prey species, as well as some sort of interaction information on them. Or it can be extremely broad in terms of general ecology. For example, intertidal rock pool ecology, you would sample that entire rock pool, or the invertebrates or the fish species. You would probably also collect some physical information such as temperature, size of the rock pool, depth of the rock pool, etc. And then you would have a general understanding of the ecology of that rock pool. So this one really, or ecological extent really, focuses you on what parameters you need to collect, whether it be biological parameters in terms of sampling individuals, or physical parameters in terms of understanding the environment that your sample species is within, so that you're able to connect environmental or physical attributes to behavior within your sample species. And really the focus for that is just have a clear understanding of what parameters need to be collected or observed. Once you have decided on your spatial extent of your study area, the temporal extent or the time frame at which you want to collect samples, and the ecological extent in that the parameters that you require information on to be able to make ecological inference later, the next step is to actually define a sampling unit. So the collection of sampling units makes up the sample of the entire population. So the sampling unit is essentially the highest resolution of our ability to make inference. It's the highest resolution information that we're going to get. So we will be able to aggregate these sampling units and make resolution or make inference at a lower resolution, but we'll never be able to get a higher resolution than the sampling unit. So what's important in this is that all parameters must be able to be measured at this scale or this resolution. So as an example, I'm going to walk you through a study that I was involved in where we decided to make the sampling unit a square 250 by 250 meter grid cell with 5 subsamples within that grid cell, biological subsamples. So it was a study where we were looking at the Benthic environment offshore of the Amatola, and we were using a remote operated vehicle, and we were flying transects across the sea floor. Each transect was between 1 and 1.5 kilometers, and the footage was continually being, or the video was continually being shot onto the benthic environment. What we then did is we overlaid the grid, and we got 5 equally spaced points on the ROV transect within a single cell. We then extracted the video footage, a single picture from each one of those 5 points, and we analyzed the benthic organisms that were in that particular photograph using CPC. We will also then average the physical parameters within that cell. So we took the average depth, we took the average profile of the substrate, and we used Katami to get an average understanding of what the substrate was, whether it be rock, reef, sand, etc. The reason we did the averaging is because we weren't able to get exact locations of where these photographs were because of the way that the ROV was flown. So we had to aggregate to a 1/4 kilometer grid cell. That then became our sampling unit, which you can see at the end there. Each one of these colored dots was a sampling unit with 5 photographic subsamples within that, as well as aggregated physical properties of that unit. A question that's often asked is how many samples are required, or what should my sample size be? And it's a very difficult question to answer prior to actually sampling. There are methods to calculate what your sample size should be once you've done some sampling, and I'll get to those. But prior to sampling, it really is subjective. It cannot be accurately calculated, but there are a couple of things you need to bear in mind. The first is that our ability to estimate population parameters precisely is directly related to the number of observations we have, or the sample size that we have. So the more samples you collect, the more precise your ability to make inference is going to be, bearing in mind that obviously there are logistical constraints to the number of samples you can collect. But the second thing to bear in mind is something called sufficiency. So if you are implementing a stratified sampling design, you need to have a sufficient number of samples within each stratification to be able to run models. So that's also something we need to bear in mind if you are implementing a stratified sampling design. So if you say, for example, stratify your area according to depth and reef or sand, if you don't have any deep reef environments, you're going to struggle to, or samples from deep reef environments, you're going to really struggle to do any sort of analysis after that. But once you've collected some information, there is a means of understanding whether you have enough samples to make decent amount of inference. And there are 2 methods for single species abundance estimates. There's something called a power analysis. A power analysis essentially determines the sample size required to estimate or to be able to detect a percentage change in a population given a predetermined power. In this case, we were looking for, with the diagram on the left, we were looking for to be able to get, or to be able to determine a 10% change in a population over 5 years with 80% power. When you're looking at multi-species, we use something called a species accumulation curve. And essentially what that's saying is, if you add more samples, how many new species are you going to encounter? And if we get a situation where the species accumulation curves levels off, in this case for coarse sand and to some extent for roadless, then you've sampled enough, because by adding more samples, you're likely not to encounter a new species, which means that if you're looking at a multi-species or looking to understand what species exist in an area, you've sampled enough. So after you've determined how many samples you should collect or what your sample size should be, the next obvious question is, well, how do I distribute my samples across my study area? And there's three methods of doing this. The first is random. I'm not going to talk about that. It's pretty obvious. It's completely random. It's generally not employed in ecology because it's quite hard to remain completely random. The two methods that we generally employ are stratified or systematic. So a stratified random sampling design is essentially stratifying your study area according to known physiological habitats. So an obvious one is, in this example, we have sediment or low profile reef or high profile reef. We know where the reef is, we've mapped it, and we're going to say, or stratify our sampling according to that. And in this case, you then decide to put X amount of samples on each one of those habitats. The stratified random sampling is preferable when the variance within a stratum, so say the variance within high profile reef is less, or the variation within high profile reefs is less than the variation across all stratums. So that's when that is a preferred sampling design. And the second is the systematic sampling, which I've kind of shown you in the example where we overlaid a grid across our sampling area and used a grid as our sampling unit. This can improve your precision because it's essentially random to some extent, in that your starting point of your grid determines where all the other points of your grids are going to be. But it really needs to be well designed, and particularly the spacing of the grid. How big is a grid needs to be well designed. The last concept I'm going to talk about, which is important to sampling design, is something called spatial autocorrelation. The majority of studies I've known, ecological studies, simply neglect to address this, and it has consequences, particularly in terms of sample independence. So in 1970s, a geographer called Tobler described spatial autocorrelation in a very simple sentence, saying, everything is related to everything else, but near things are more related than distant things. In the map below, you have GDPs of all the countries in the world, which are obviously a result of numerous things, whether it be economics, political agendas, etc., historical backgrounds. A GDP is a result of numerous parameters. But despite that, a country's GDP can be pretty accurately determined by simply looking at the GDPs of the countries which surround it. And that is exactly what spatial autocorrelation is. There is information in the surroundings. So in 1993, a very good numerical ecologist called Legendre wrote a paper titled Spatial Autocorrelation, Trouble or New Paradigm. And what he simply said there was that spatial autocorrelation is the fact of life for ecologists, and in the past it's been seen as a statistical nuisance, but really, if we deal with it appropriately, it can be a great source of ecological information. But when dealing with designing a sampling protocol, the one thing you must bear in mind is that when there is spatial autocorrelation, it violates statistical assumptions, particularly that of independence. So you must bear in mind that if two of your sampling units, neighboring sampling units, are autocorrelated spatially, they cannot be seen as individual or independent sampling units. So just bear that in mind. We can see spatial autocorrelation at the community level. This results from our study in Tsitsikamma, where we saw communities grouping out and being spatially autocorrelated with nearby cells likely belonging to the same community. We also see it at a species level. This is abundance data of Dageraad at Tsitsikamma. And what we see is that grid cells next to each other had very similar abundance estimates. So there was some definitive spatial structuring here, and that needs to be accounted for when you are modeling larger ecological processes. So you can quantify spatial autocorrelation using a correlogram, which is shown here. This is a correlogram of abundance data of Dageraad and Tsitsikamma, which I showed you in the previous slide. And essentially what a correlogram does is it estimates the average correlation between sample sites a known distance apart, and it keeps increasing that distance. So what we see here is that there is a high correlation between sites within 300 meters of each other. Our grid cells were 150 meters in this case, so essentially the neighboring grid cells were collecting redundant data and there was a lack of independence between them. So what we eventually ended up doing was doubling the size of our sampling units in this particular research study. So understanding spatial autocorrelation really gives you a good idea about the independence of your sampling units, as well as a good idea of ensuring that you don't waste resources collecting redundant data because they are spatially autocorrelated. I think I've run out of time, but hopefully I've been able to give you a very brief introduction as to how to go about setting up a sampling design. I think the take-home message from me is really a well-designed sampling protocol will save you a lot of frustration when you come to do analyses. So take your time in understanding the intricacies of your area, how many samples you may require, how you're going to distribute those samples, and consider spatial autocorrelation. If you do these things, you will save yourself a lot of frustration when it comes to the actual modeling and inference side of your study. Good luck and questions.
Oh, that's brilliant. Thank you very much, Denham. I'm very glad to have had this. This is actually an excellent primer for everyone, and I hope people are taking notes, because as you say, these are really important questions. As I see that there aren't any questions in the Q&A box at the moment, but I'm going to take advantage of that and just see if I can satisfy some of my my own questions, the things that I've come up with, because certainly the points that you've been talking about are absolutely things that we deal with in trying to do our surveys and design our studies. But I wonder, especially in terms of species accumulation, over which scale would you typically look at that? Because you can look at it over the entire study, or as you had in your plot there, according to substratum type and so forth. Is there a sort of a typical way of doing that? I suppose it depends on your question, but in terms of the study design and validating that, is there a particular scale that you would want to look at it relative to perhaps, well, your sampling unit, but also your sort of scale of processes or habitats and that kind of thing?
Yeah, thanks, Charles. Can you hear me?
Can now, yes.
Okay. Yeah, good question. So again, it's very dependent on the question you want to ask in your study. But ideally, if you have stratified your sampling according to, for example, benthos type, whether it be sand or reef, for example, you would want to do a species accumulation curve based on that stratification, because it will essentially provide you with information as to where you are lacking data. So what we find is when we do that kind of study, before we finish sampling, if we did a species accumulation curve, we could change our sampling protocol slightly. So we normally find that we would have enough information on reef or rock benthos, but we would probably want to collect a bit more on muddy or sand benthos, because the variability within that data is so high, because you're not getting many observations. So it's good to do that sort of stratification, and then maybe if you've got one more or two more sampling events coming up, you can kind of prioritize a substrate where you don't have enough data. So it's an ongoing process that you can then fiddle and manipulate with your upcoming sampling.
Oh, fantastic, thank you. That's great. I see now we've got a few questions here. And so we've got one here, yeah, great talk. If I wanted to go out and confirm or ground truth, perhaps nursery areas or preferred habitat types of a commercial fish using cameras, how would you suggest I structure my sampling design in terms of random or systematic, etc.? And what are the sort of important factors that I should consider?
Okay, so if you're dealing with commercial fisheries, obviously the one thing you want to consider when you're setting up your sampling design is what are the limitations of the commercial fishery. And what I mean by that is, if you are looking at a trawl fishery, you certainly don't want to be sampling reefs, because that's not where the trawl fishery is going to actually operate. So I would first get some idea of where your fishery operates spatially, look at the parameters such as benthos that they're operating on, and then stratify according to those. So for example, a trawl fishery would be stratified only on unconsolidated benthos, and likely based on the different depth profiles that they would be sampling. So then you can bin your depths. Say if your depth range is 200 meters, you can bin it into 3 or 4 or 5 bin categories, and then distribute your sampling effort amongst those depth categories.
Okay, another one here, thanks. The question, how is spatial connectivity used in site selection for MPAs? That may come up later, but if you have something to input there, that would be great. As it relates to what you've, the points that you've spoken about. Yeah,
connectivity, spatial connectivity is something that is considered in well, first of all, you need to first understand what your MPA is, why you would want to put in MPAs. If it's something like the Table Bay National Park, where you have a lot of small MPAs that are specifically focused on reef sort of habitats, then the connectivity you would quantify the connectivity of the fish, the reef fish within those habitats, and their movement. So it's a difficult question to answer, but it all really depends on what your MPA objectives are. If there's a specific species that you're trying to protect, and if they are a group of species, and if they are, what is their spatial distribution and their ability to move. Then if you understand all those things, then you can start looking at connectivity between MPAs or between MPAs and exploited zones.
Fantastic, thank you. Okay, so we've got another question here from Paris. Are there any sources or manuals that can help get started with estimating adequate sampling effort? So for instance, power estimates and power analysis and so forth.
Yeah, I will put those resources up. I mean, I don't have them in front of me, but I've definitely hand them over to the ones that I believe to be useful. Zur is a good one. Like I said, Legendre, when you spatial autocorrelation, for me, that was really a study that highlighted the effects of just completely ignoring space in terms of ecological monitoring and ecological project design. And I think that's important. And they've also, Legendre have a book called, I think it's Numerical Ecology, which is also a really, really good book. It's something I would look into. But I will definitely hand those over to the panelists.
Thanks. That is good. Yes, I do know those and very useful. So another one here, should it be standard practice to present sampling efforts, so that, you know, species accumulation plots for biodiversity surveys when presenting results, so that that can be gauged?
Yes, in some aspects, well, at least in some way. I mean, I know that there's a move when you towards making papers shorter and shorter, but at least I would present them in some appendix format. It just gives the reader a lot more confidence in the data's ability, or the analyses that have been done on the data, and the ability to gain inference, any sort of inference from the data. So for me, it's very important as a reader to understand the number of samples, the distribution of samples. You don't need that in-depth information, but you do need an overview to get a sense of how much confidence you can put into the analysis and the data, the results that are coming out from the analysis. So I think as far as you can, be completely open about that. Put it in appendices, note it somewhere. It definitely makes a difference. It gives readers a lot more confidence in your outcomes.
No, great. I think we've got time for just one more quickly. It's an interesting one here where the person's asking, is there a concept of temporal serial autocorrelation, and how may this data, well, influence data collection or design? Yeah.
Yeah, there is. So temporal autocorrelation is also a big thing. The difference, and why I didn't bring it up so much, is because generally what we do in ecology is to try look at changes over time, whereas the space aspect of it is ignored. So the autocorrelation within time can be considered if you have something like an AR1 model. There are means of doing it, putting the autocorrelation structure into your residuals or accounting for it in your residuals. But by virtue of the fact that we normally try and model for change over time, it has a little less influence than the spatial aspect, which can be completely ignored. But it is, if that is something you are interested in, have a look. And at least if you're doing projects that last over a year, the bare minimum you should do is try account for seasonality, whether it be by noting within your models the month or the season in which they were collected. So that is standard practice. Is if you're analyzing data across the year, at least try account for seasonality to some extent deal with that temporal autocorrelation.
Excellent. That's fantastic. I think we've got one more here, which is, do you have any advice for how spatial autocorrelation assessment could be incorporated into citizen science data collection programs?
Oof, that's a very specific question. So for me, the spatial autocorrelation aspect of it is not how to incorporate it immediately in your sampling design, but is to be aware that it exists, to know how to treat it. If because in every single sampling protocol, there is a spatial aspect to it. So be aware it exists, know how to treat it, and understand that by virtue of the fact that you can tell the difference between two samples and you know the distance between those two samples, there is information to be gained from that. And I think that's important. You have two samples, you can quantify them, you can know exactly how they differ, and you know the distance between them. That in itself is a source of information that a lot of people don't use.
No, that's yeah, that's a good answer actually. That's you know, something that just struck me when you were talking there about knowing the distance from your samples, where you have, like you had with the case of the example study from Amatole, where you have ROV imagery and stuff where you may know a start and an end point, but you don't know exact locations of images. I suppose that always means then that you can't look at that aspect when you don't know those locations. You have to aggregate, like perhaps like you said there, or that you spoke about, in order to do that.
Yeah, so there are obviously constraints to every sampling procedures, and that was one of our major constraints, is not knowing exactly where our photograph specific information came from. But we overcame it, and by aggregating that information, and in doing so, you probably get a more holistic picture. The key there is at what level do you aggregate at? And the aggregation needs to be across all parameters. So normally what happens is there is a parameter which is like your minimum quantity that you have to, that is like, we can't collect that information at a finer scale, so that's the scale we have to aggregate at. And it seemed to work out for us in that particular case. And again, it's just having the foresight to understand the spatial autocorrelation, and at what level can you make the most of that spatial information. If you go too fine, you may hinder yourself. If you go too broad, your results may be a little bit, or at least not reflect the resolution that you want.
Sure. No, thank you. That makes sense. Okay, I think that's all the questions for the moment. I think that was fantastic, very worthwhile, and I hope that's going to help people put into place both the pre-study concepts that they need to think about, and sort of checklist of what you need to consider, and the post, like dealing with the autocorrelation, and yeah, knowing what to do there. So thank you very much, Denham. That was fantastic.
Thanks, Charles.
Great. Okay, I think we're having a poll coming up just between now and the next speaker. So just go ahead and answer that. And we will continue with Dr. Alison Kok after that. Okay, welcome back, everyone. Very interesting there. I see that most of us have been affected by COVID, especially, I suppose, long-term studies. We're going to now go over to Dr. Alison Koch. Sorry, I'm jiggling my screen here. But yeah, so we're going over to Dr. Alison Koch, who's a marine biologist with Cape Research Centre, South African National Parks. And she implements and coordinates research and monitoring programs in marine protected areas and translates that into management and biodiversity actions. Alison has co-established the Save Our Seas Shark Centre in Kalk Bay, South Africa. And the vision of that is sustainable coexistence of people and sharks. So her interests include conservation biology, top predator conservation, especially protected area effectiveness, biodiversity monitoring, behavioral ecology, and human wildlife conflict mitigation. So welcome, Alison, and yeah, looking forward to hearing from you.
Good afternoon, everybody. My name is Dr. Alison Koch, and I'm a marine biologist at the Cape Research Centre for South African National Parks. Here I lead research and monitoring on marine protected areas, and I provide scientific advice for evidence-based decision-making and adaptive management. I've worked at SANParks for 4 years, and I love the interface between science, management, and policy. MPAs that I work in extend all the way from Cape Town to the Northern Cape and include the Table Mountain National Park Marine Protected Area, Robben Island MPA, West Coast National Park, and Namakwa National Park. Today, I'll be sharing some tips on designing marine protected area monitoring plans, key considerations, and prioritizing the legislated objectives and the threats or pressures that are to the MPA. Today, I'm focusing on marine protected area monitoring plans, but the concepts of this presentation are applicable to most other monitoring plans as well. Thus, for today, I want to just explain what a marine protected area is. It means different things in different countries, but generally speaking, it is a designated coastal or ocean space that is legally protected and managed to conserve valuable and sensitive ecosystems and species for the benefit and service of people. In South Africa, we have a network of 42 marine protected areas that cover approximately 5.4% of our exclusive economic zone. This network was established using systematic conservation planning to identify key biodiversity areas representing all the ecosystem types in South Africa and those that really needed protection. Each marine protected area, however, has got a specific set of legal objectives and its own set of pressures and threats that need to be understood for that marine protected area. Also in South Africa, marine protected areas are usually zoned to further try and balance social, economic, and ecological needs. Most marine protected areas in South Africa have got a combination of a controlled zone and a restricted zone. Controlled zones allow for limited extractive activities, while restricted zones don't allow for any extractive activities and only allow for low impact activities. In general, large-scale activities like trawling, longlining, mining, and aquaculture are not permitted in any of the zones in the marine protected area. Monitoring plans really need to try and cover all the zones of the MPA, and where applicable, also to cover outside of the MPA so that you've got comparative areas for your monitoring results. As mentioned, each marine protected area has got its own set of objectives in why it was proclaimed. In South Africa, these are usually formalized through legislation. However, some general objectives of MPAs include maintaining or improving biodiversity and productivity, maintaining the ecosystems that the marine protected areas offer, protecting rare and critical habitats and species, providing fisheries spillover, protecting geological features, as well as cultural assets and values, providing opportunities for recreation and tourism, providing baseline environmental information of marine spaces to look at the impact of global change, and then also providing opportunities for education and research. Each marine protected area also has its own specific threats or pressures facing it, but in general, these include things like resource and unsustainable fishing. Climate change is increasingly becoming a major pressure we need to consider. The loss of top predators, invasive alien species, habitat destruction and mining, and pollution. To give you an example of how these might be applicable to different marine protected areas, Namakwa National Park Marine Protected Area is situated in the far Northern Cape. This area is heavily utilized by mining. And so the main pressures facing this marine protected area are from mining. Climate change is also a big pressure due to the location of the marine protected area. Alien invasive species are also an issue there. But for example, because of its remoteness, overharvesting of is not a primary pressure for this marine protected area. So just stressing once again that each marine protected area has its own set of threats and pressures that need to be considered. So why do we need to monitor marine protected areas? Monitoring is an essential component of resource and adaptive management. It provides science-based information to guide key management decisions, and these include the prioritization of conservation strategies and conservation targets, ensuring that there's proper allocation of resources and staff, determining whether or not the marine protected area meets its intended objectives, and increasingly there's pressure to demonstrate the benefits that marine protected areas have to people, particularly those that live adjacent to the marine protected areas. And it's critical to note that attempts to quantify the effect of marine protected areas or marine protected area networks are usually hampered by a lack of well-designed monitoring studies. So where do you start in developing your monitoring plan? A good place to start are the legal objectives. In South Africa, our marine protected areas are proclaimed through government gazettes, and these usually have objectives or purposes set out in the government gazette that have involved the input of stakeholders. So with the new PAKISA MPAs, around 20 that were recently gazetted in 2019, each of these marine protected areas have specific purposes. And this is an example here of what this looks like. For Robben Island, for example. So you'll have a list of specific purposes for that marine protected area. And it's really important that your monitoring plan talks to these objectives. The next place to look for areas of importance and prioritization of your monitoring plan is at the park or the MPA management plan. This is an example here of the West Coast National Park Management Plan. It's got very high level objectives in it. I don't expect you to read the text, but this is just an example of specific high level objectives with actions and role abilities of those actions. So it's really important to have a look at your MPA management plan. This process is also usually done with stakeholders, so you get stakeholder engagement and input into what is important, what needs to be prioritized, and what your stakeholders think are important things to monitor. Also getting started, but before you embark on your monitoring, it's really important to understand what the knowledge base already is of that marine protected area. So, for example, in sand parks, we have state of knowledge reports. These essentially are a review of the existing knowledge, scientific, indigenous, local community knowledge that is available about your marine protected area or your national park. And this kind of foundational knowledge includes the location, area, boundaries and buffer zones of your marine protected areas, the managing body, the type of management, legislation, jurisdictions, and socio-cultural structure of your marine protected area. It also lists your types of resource use, as well as users, your ecological, your socio-economic, your cultural, and your traditional information. It should also include a list of stakeholders, including potential collaborators for your monitoring. And then also it should contain inventories of species, such as species lists, cultural assets, and so forth. So it's really a good idea to collate and review existing knowledge for your marine protected area, so that you have got a good baseline of where to start and you don't start reinventing the wheel for your monitoring. There's a lot of sources of information for your state of knowledge. You know, once again, you need to look at your legislative objectives, your management plan. In South Africa, we have what is called a management effectiveness tracking tool, or a MET assessment. There's a lot of fantastic information in these. These are generally done once a year by the park or the MPA and contain a long history of actions and progress with regards to the MPA. Of course, the academic literature, there's often quite a bit of research that's already been done in that area. To access that academic literature, if you don't belong to an academic institute, you can always email the author. They're always happy to email their papers, collaborate with others. You might be able to get an affiliation with an academic institution that gives you access to academic papers. And of course, the internet through Google Scholar is a really good source. Really importantly, talk to past and present staff, your rangers, your managers, your scientists, to gather information on the marine protected area. Site visits are essential to better understand the region before you set up your monitoring plan. Park and MPA forum meetings and past minutes are also a valuable source of information. Engage in your external partners, your stakeholders, other people that have worked in your MPA before. You could also host or participate in stakeholder meetings and try and include as much stakeholder engagement in developing your monitoring plans. And then, of course, the WIO network, the Western Indian Ocean Protected Areas Network, that was recently launched, is a fantastic source of information that you can also access. Now that you have your state of knowledge and understand what is already known about your marine protected area, including its ecology, socioeconomic and cultural attributes, you can start to draft your monitoring plan further. Your monitoring plan should really include the following. It's not limited this, but you can include the following: your state of knowledge. If there was an existing one, you can update that. Your specific monitoring objectives, your indicators, which are really important in how you're going to meet those objectives, the methods that you're going to be employing to do the monitoring, your sampling design, which I'll be talking about a little bit more. You need to decide on your monitoring frequency, your survey team. Here's a picture of our West Coast National Park baited remote underwater survey team, the necessary equipment that you need, and the budget that you'll need as well. We really try from scientific services to work closely with our park staff and stakeholders when implementing our monitoring. So I would definitely encourage you all to get more participation in your monitoring for your marine protected area. Of course, there are a range of monitoring types. You've got short, medium, and long-term monitoring goals. The monitoring types include ecological monitoring, such as your biodiversity, your habitats, your species. And then we tend to forget the socioeconomic monitoring that's needed as well. But increasingly, this is becoming very important for marine protected areas. And that's to better understand the users of your marine protected area, such as your fishers and your tourism operators, for example. And then, of course, monitoring of your governance, which includes your law enforcement. We've got specific management effectiveness tracking tool to do that, but there are also other processes within organizations to look at the impact of your governance. And then typically when it comes to marine protected areas, ideally you'd love to have some baseline surveys. These are done either before proclamation of the MPA or very soon thereafter, so that you get a good understanding of the characteristics and attributes of your marine protected area before it was proclaimed. And then from there, we usually want to implement long-term socio-ecological surveys that are continued through the existence of the marine protected area. Really importantly, we need to generate data that are adequate for answering the questions that motivated the marine protected area in the first place. This is a really critical component of the design of your monitoring plan. Of course, there are literally thousands of different kinds of data that you can collect. And this is why it is so important that the data you collect talks directly to your objectives and the pressures and threats that are applicable to the MPA. It's essential to prioritize given how many variables one can actually collect. Across the different categories of data, you've got your biophysical data, which includes your water temperature and water quality in some cases. For example, this is particularly applicable to the Table Mountain National Park, which borders a large urban city. You've got your ecological kinds of data, your habitats, your species. For example, very applicable to this workshop, your benthic invertebrate abundance and diversity, your fish abundance and diversity, seabird breeding pairs, for example. And then, of course, your socioeconomic data, the people and the use of the marine protected area. So these things include lists of activities that occur inside the marine protected area, catch per unit effort, for example, the number of fishing permits, your stakeholder list, and then your governance. So the kinds of data that you could be looking at monitoring over time is your staff capacity, the number of operations, the number of patrols, and so forth. And then, you know, increasingly, you need to think about how technology and automation can help you with your various monitoring goals. Of course, as practitioners working in the marine field, I'm sure you are well aware of the limited resources and capacity that we have in this environment, particularly for monitoring. So when designing the monitoring plan, I strongly advise that you prioritize monitoring that addresses more than one objective, and you also look for collaborative opportunities. So for example, in South Africa, there's a National Rocky Intertidal Invertebrate Survey, and this takes place across several marine protected areas. And it covers monitoring the habitat type and then also several key species as well. In Namakwa, for example, we collaborate with the Department of Forestry, Fisheries and Environment, Oceans and Coasts Branch, the Department of Environment of the Northern Cape, as well as the South African Environmental Observation Network. And this particularly rock ashore monitoring that we do speaks to the main gazetted objectives of Namakwa MPA and its main threats. So it really talks to monitoring the impacts of mining, climate change, and alien invasive species. It also can look at harvesting, but that's not one of the main pressures that we have. Rocky shore monitoring, for example, is also low cost, and it's really fairly easy to train people in this technique. And so you really want to look at monitoring programs that maximize your capacity and your resources and opportunities for collaboration. The sampling design of your data collection and your monitoring plan is critical. It's often an overlooked component of the monitoring when setting it up. You really don't want to dedicate time and resource use to collect data, but then 1 or 2 or more years down the line, you find that your data have not been adequately collected to answer your questions. And in fact, you sit with all this data that's been collected, but it can't be used. So luckily in this WIOMSA workshop, there are several presentations covering data collection as well as sampling design. So I won't go into that in too much detail, except to stress that your sampling design really depends on the key questions and hypotheses you want to ask and address. You really need to implement the appropriate methods and analyses. And then most importantly, don't be afraid to seek expert guidance. So for example, statistics guidance on exactly how your data are going to address the questions that you have. It's really important to try and do this before you set up your monitoring and go and collect your data. So I really strongly advise you to get that expert guidance if you need it. Another critical component of your monitoring plan is proper data management. While you're sitting here listening to this talk, I want you to think about the data that you currently have collected and is probably sitting on your laptop. What would happen if your hard drive failed today? Do you have another copy of your data? If something had to happen to you today, would others have access to your data? Would they be able to understand your data? Would they be able to continue collecting the data in the same way that you have? This is where the data management comes in really importantly. And data management plans should really be a strong component of your overall monitoring plan. Your data management includes several components. Your data management planning, it should really cover the legal, policy and ethics, as well as the strategic objectives. So for example, if your monitoring includes attaching GPS loggers to birds, you probably need some ethical clearance. What are the references to those ethics that others can be aware of, and when you need it to publish your work, or when others need to follow your work. And then your data management plan should also really talk and connect with your strategic objectives of your organization. It really needs to include information on data capture, including your metadata. So it's really important that you include metadata in your data so that people can understand what each field means and exactly how it was captured. And then your data management plan should also cover how your data is going to be stored, where it's going to be backed up, and increasingly we are using cloud-based systems to ensure that our data is preserved in the long term and accessible for many years to come, because you really want to have the time and the effort that you've gone into collecting data be available for people in 20, 30 or 40 years time. That is the ultimate goal. And then you also want to include information on who has access to that data, any acknowledgements that need to go with that, any funding acknowledgements that need to go with that, and who they should get in touch with if they want to access the data. I would really like to emphasize that monitoring without producing recommendations and without having interventions is useless. It just uses resources. And so this is an example I wanted to show you of the numbers of breeding pairs of African penguins over time. African penguins are the only endemic penguin species found in South Africa and Namibia and are currently at risk of extinction. So is the idea for us here to just monitor the population until it's gone? As you can see here, there's been a steep decline in the total population as well as in South Africa. And the question is, do we just want to monitor it into extinction? Well, no, we absolutely don't. We want to use this monitoring information to make recommendations for interventions to stop the decline and to eventually reverse that decline. And this is currently a process being actioned at the moment. Therefore, it is essential that our monitoring work does not just sit on our computers, but that it feeds into management. And we call this closing the monitoring loop. It's a really good idea to identify these steps in your monitoring plan. This is a schematic drawing from a publication that's currently open access and available in Kudu. The details are on the slide. Essentially, what you want to do is you want to identify your management concern. And in this particular example, the concern is around recreational and subsistence fishing occurring within the Garden Route National Park. And the starting point was that there was little to no information available on the efforts involved, which species were being caught, and where management needed to focus. And so the next step really is to prioritize where the monitoring should take place, where the effort needs to be concentrated, and to develop key questions and hypotheses around this management concern. So then you want to implement your monitoring and some of the data that you want to collect to be able to address your question. Importantly, you want some social and economic information. You want to know the total effort. You want to know the effort type. You really want to understand what the user's knowledge of the regulations is. You really want to understand what proportion of anglers are complying or not complying to the regulations. And you want to understand what are the reasons and the motivations for fishing in the area. Some of the ecological information that you'd like to understand is looking at overall catch per unit effort, for example, targeted catch per unit effort, and then also what species are being caught and which sizes are being caught. And then you also want to look at your organizational or institutional kinds of data, your levels of non-compliance, the number of patrols and inspections that you do, and your adaptive management processes. Once you've got and collected that information in your monitoring, it's really important to give this back to management for feedback. And so these could be places such as science management forums. In sandparks, we have got formal science management engagements, your annual reporting, and really the analysis of what you've uncovered through your monitoring. And so what are the kinds of management interventions or actions that could be implemented from this monitoring work? You could look at whether your signage is appropriate. You could look at whether your knowledge and awareness information is appropriate. And if not, you could design brochures and field guides that talk to the knowledge gaps or the information collected from your monitoring and your research. You could look at having more workshops and training for your staff if needed around fish ID, for example. And you could also look at perhaps changing your enforcement with an emphasis on, you know, the importance of maintaining your bag limits and your sizes. And once you've got this information and you've identified management measures, you feed that back into your monitoring and you continue to revise this process to be the most effective. And then, of course, really importantly, I want to stress the communication of your work and the importance of communicating your work broadly and to your stakeholders. It really is important to showcase your work. You could do this through your annual research reports, on your websites, other people's websites, through blogs, through doing presentations. Online presentations have now become fairly easy to do. You could write popular articles and so forth as well. And then before I end off today, I would really like to go over some of the main pitfalls that I've seen when implementing monitoring plans. Lessons learned myself from falling into the pit of some of these, and then also some solutions. So again, monitoring without interventions is of no use. You really want to prioritize your legislated objectives and your management plan actions and those threats that are applicable to your MPA. You don't want to monitor the easy stuff or just the stuff that is of interest to you. This is really important. You want to prioritize once again, and you want to understand that your monitoring talks to a much broader picture than just the stuff that you want to monitor. Also, don't start off by being too ambitious and wanting to monitor everything. There's so much to monitor, but remember to prioritize. And ideally, you want to prioritize monitoring that speaks directly to the applicable threats or pressures to your marine protected area. And then there's so many different monitoring methods. You know, for example, to monitor fish, you can use, you know, catch per unit effort from angling. You can use baited remote underwater videos. You can use seine nets. You can use scuba counts. So really do your homework about the best method that is applicable to your marine protected area. And then don't duplicate efforts. Really do your homework and collaborate wherever possible. And finally, and most importantly, have some fun. It's an absolute privilege to work in the marine science field, and you really need to make some time to get out there, away from your computer, to remember why you do the work that you do. Thank you so much for listening, and I really hope that you enjoy the rest of this workshop.
Fantastic. Thank you so much, Alison. That's really, I like the bit about getting away from your computer. That's an important one, and a fantastic, like, holistic look at marine protected area design and all the aspects that come into that. I see that we have a question here. Is there a software that allows you to combine all the different data layers used in MPA design, planning processes together? Something that you use, perhaps?
Thanks very much, Charles. Yes, there is free software called Markzan. That is available to input your different data into. Essentially, it really helps to, it's a conservation planning tool, and it really helps to identify areas that have got specific habitats, specific species. So you can have your different data layers, your different habitat types, perhaps species distributions. There's a range of data that you could feed into Marxan into these layers. And essentially what you can then look at is the percentage of species that are in a particular zone of the MPA, or a percentage of habitat that's covered by a restricted zone versus a controlled zone. So that software is freely available, and that's Marxan. I can get a link and type that into the chat box once I'm done here.
No, fantastic. Of course, Marxan. That's great. We have another one. It's basically about the person agrees that ecological monitoring requires long-term funding. But is there any technology advancement like CCTV that could be installed in selected sites to obtain continuous ecological data? Yeah.
Yeah, I mean, increasingly technology is providing solutions to monitoring lots of information. And for example, there are CCTV cameras going up in places like seabird colonies to monitor breeding pairs as well as behavior. It's been used to monitor key access areas, for example, like slipways, where you want to get an idea of how many fishermen or fishers are going out in their boats to look at the amount of effort in terms of fishing that's happening. And so they're increasingly being used. I think the bottleneck comes again with analyzing that data. And, when we're using imagery such as videos, there's a lot of video to watch. And it's coming up with automated tools, I think, to match those technological advancements now, to ensure that you're not just monitoring for the sake of monitoring, but you're actually using that data in a timeless way, and it's one of the challenges that we still face, is making sure that once you've collected your data, you're doing your in the field monitoring, that you actually analyze that in a way that's timely and can then feed back into your management recommendations.
No, absolutely. That's a really excellent point. And yeah, it's all very well collecting reams and reams of continuous video data or video samples, but yeah, having the ability to process that is essential. I have the next question here, still in the Q&A, but I do see that there is a hand raised. One of the most challenges existing to most MPAs in the WIO region, especially in the side where community protection is involved, is where the restriction zones are created and the reaction of the community is mostly negative, as there'll be complaints about loss of income and so forth. What should be addressed in terms of the management aspect before establishing a restricted zones? Where you have those communities that are highly dependent on income from the zones.
Increasingly, this is such an important question, with the establishment of new MPAs, particularly through the PAKISA process, Dr. Kerry Sink has a lot of advice for including stakeholders and the importance of including stakeholders in the design of the marine protected area, including its restricted zones. Even in your monitoring plan, it's really important to be as participatory as possible, so that people feel heard, because often the problem comes down to people not feeling heard in the development of marine protection across the country. I think it's a global problem, not only in the WIOMSA region. So stakeholder engagement from the very, very beginning is an important lesson that's been learned in the establishment of marine protected areas. And that really takes place across a range of scales. And so before you even proclaim your marine protected area, you need to have stakeholder engagement. And then even once it's proclaimed, you really need to keep communicating to your stakeholders why it was designated into a restricted zone or to a controlled zone. That's where your data comes in and plays a very important role, so that you can actually show which habitats and which species are being protected by which zones and why, because that's a really important part. And then again, your monitoring feeds into being able to show your stakeholders why it's important to keep these areas closed, for example, and to demonstrate potential benefits to the community that might have been impacted by no longer having access to a particular, stretch of coastline. And so the key message is stakeholder engagement at every possible opportunity that you can, so that people feel heard and can be part of the process.
Okay, great. Thank you. Yes. I think I'm going to move to a question. There was a hand raised. Can that come through? No. Okay, I'm just going to, while that's being sorted out, I'm just going to go back to the Q&A box there. And there's a comment here. Thanks, Alison, for the presentation. I learned a lot. Could you highlight a few examples of cultural assets and traditional knowledge that you have used in your management plans or planning? So cultural goods and services are mostly ignored with MPAs, and I was happy to hear that South Africa has tapped into this.
Yes, so the coastline is a rich source of cultural heritage and knowledge, and it's certainly at risk of being lost, not documented, perhaps allowing for certain activities that influence those. So for example, across many of our marine protected areas, there are what we call these middens. They are essentially places where indigenous people used to forage and feed, and so you can find remnants of bowls and items that they perhaps fed on, different shell types, and so they're really important cultural areas, and they're often not very obvious. And so the first step is really to do an assessment of what cultural assets you have got in your marine protected areas, and that can include things like shipwrecks, caves that were used by indigenous people that might still be used, and really just first of all have an asset list of what you have with the specific location of that, and then that information can be used in making any kinds of management decisions around where to locate a new slipway, or where to build a building, or where not to, or where to direct your tourism activities. Perhaps it's something that you want to highlight and provide access to, but then you need to set up the right infrastructure and make sure that it's not destroyed in any way and is there to be enjoyed. So yeah, the first bit would be to identify those cultural heritage sites and ensure that they are included in your management plan, and that management is aware of them.
Excellent, absolutely. I think very much or often underappreciated or under-considered aspects of MPAs, and just in general, really. I think we had the hand raised from Bernard. Are you able to unmute yourself? Perhaps that's the problem. Bernard, if you can unmute yourself to ask a question, go ahead. I'll just give him a sec. Hi, yes, go ahead.
Yes, I have no question, please. It was a problem with my machine here. So I'm okay.
Okay, no problem. All right. I think that we're pretty much on time here. So I guess just to summarize, thanks very much for that great talk. And I think we will have a poll coming up. But I think that's having those two talks has really drawn through together the two aspects that actually relate, having the correct design and keeping that, keeping everything aligned so that you get you meet your objectives, even over the long term. So thank you to both of you very much for those informative talks. I think that's, I know I've learned a lot, and I hope they've been useful for everyone. So please go ahead and engage with the poll. And then there's going to be a lunch break. We'll obviously continue from there. I see there's, I don't know if we can go back to it, but I see that there is another question that's come through from Alison, for Alison. So yeah, I'm not sure if you can come back in, but essentially that question was, were you able to quantify the benefits of MPAs from their beginnings? So I guess, I don't know if there's a specific example, but I suppose in the MPAs that you've been involved with.
Yes. At the moment, we have got a really exciting project on the go for the Table Mountain National Park Marine Protected Area. And the funding came outside from the Table Mountain Fund, in fact, where there were baseline studies done of the key invertebrate species. For example, your rock lobster, your abalone, your density of kelp, your urchins, and then also baseline surveys done for the fish diversity and abundance all the way around the Cape Peninsula before the park was actually proclaimed in its current state. And so we managed to get funding to repeat those baseline surveys, and we used the same methodology. And we're busy analyzing the data now with some really interesting findings. And the long and short of it is that the marine protected area is doing well in some aspects and then not doing well in other aspects. But we are able to now quantify what those are and then make recommendations for management in moving forward and how to address the areas that have not been as effective as what they should be. And then we've also been fortunate with areas like the Makwa, where there's been a lot of forethought, and it's taken 20 years to actually proclaim the marine protected area. And in the meantime, there were annual surveys done before that, and now we've got baseline information. So one of the really key things is to try and get baseline information for your marine protected area. And our main challenge is getting funding to do that. So it's expensive work, and ideally you want that kind of information for all of your new protected areas within the first year of being gazetted. But it's not always the case. So you've got to do the best that you can. But yeah, there is some really nice examples that are starting to come out now.
Okay, no, fantastic. That's obviously, closing that management loop. I think that's what you called it, to actually feed that information up, especially where you can actually quantify real benefits and inform people and use that to, just set an example of that. But thanks very much. Really a fantastic presentation. And as I say, we're going to break for lunch and back at 1:30. All right.
Paris, for some reason we can't hear you. Sorry, if you can just check your microphone. It is unmuted, but we can't hear you speaking. I'll let you know when it comes on. At the moment, we can't hear you. Nothing yet.
Can you hear me now?
There we go. Excellent.
Okay.
Cool. Thank you.
Okay. Sorry. The joys of doing something via Zoom. I'm sure everyone has the same difficulties. So, hello everyone. Let's start again. Thank you all for joining. I hope you had a nice lunch break or afternoon break, depending on where you are based. So my name is Dr. Paris Stefanoulis. I'm a postdoc researcher at the University of Oxford, the Necton Foundation, and I will be chairing the second part of the sampling design session this afternoon. So before we start, some housekeeping. First of all, if you joined for the first time now, if you want to select your language, then please do so, either English or French, and you can do that by using the interpretation button that you can see at the bottom of your Zoom page. So select the one that applies to you. You can ask questions to the presenters via the Q&A functions. You can do this during the Q&A function, during the Q&A session, but you can do this also like throughout their presentation. If you want to like a specific question and you don't want to ask one, then you can just upvote with using the thumbs up icon, one of the questions, and then please use the chat box for technical issues only. So at the second part of the sampling design session today, the first speaker is going to be Dr. Lauren Williams. So Dr. Lauren Williams is a researcher at the Department of Forestry, Fisheries and Environment, and she has extensive experience in data management and geospatial science, specifically using geographic information systems or GIS in hazard and risk assessments. So her work involves the development of geospatial models and online interactive decision support tools to support policy development as well as decision-making processes. More recently, she has been developing data capture applications, which can be installed on smartphones to streamline data capture in the field and data management processes. So, and with that, I will leave the virtual floor to Dr. Lauren Williams.
Hello, everybody. My name is Lauren Williams, and the title of my presentation is The Application of Geographical Information Systems, or GIS, to support and standardize methodologies for long-term monitoring. Okay, so just the presentation outline, I'll be covering these primary components. We're going to have a quick overview of GIS. I understand that the audience is quite broad and experiences may vary, so I think that's important just to touch on so that we can all be on the same page as we go through the presentation. I'll be going through some GIS software very briefly, and there we're just speaking to proprietary versus open source. We'll then move on to methods and standardization, using GIS in the context of benthic sampling, using GIS models to standardize methods, some data considerations, and then some best practice tips when using GIS and different data. Okay, so in the COVID-19 context, I thought that it would be a bit interesting to start with, where did GIS all start? And of course, we cannot pinpoint a specific date in time to say this is exactly when GIS started. But we do have a very, very famous example that sort of relates to our current pandemic situation. Okay, so the gentleman you're seeing here is Dr. John Snow. Okay, and you can Google this in your own time, but it's a very, very early example of how spatial analysis was used to come to a decision to reveal some hidden data based on locality. Okay, so in 1854, this physician, Dr. John Snow, he took a map. There was a severe cholera outbreak, and he took a map and he started plotting where all the cholera cases were found, and then also where all the water sources were. Okay, and through this type of very crude mapping exercise, he actually discovered that cholera was spread by contaminated water, because it was people who were getting their water from these contaminated sources that were actually becoming quite sick. So yeah, so it's one of the few and famous examples, and I've left the source at the bottom here if you're interested to hear a bit more of the story. But I thought it was quite relevant in the COVID-19 context, and all the mapping and dashboards and everything we've seen that has spatially enabled data associated with it. So getting back to the gist of the presentation, what is GIS? And for a lot of people, it's a lot of different things. You know, it all depends on the context in which you are using it and you are applying it. If you're somebody that just captures data in the field, you know, a GIS might mean having a GPS device. You know, if you're somebody on the road that does deliveries, your form of GIS technology might be in the form of a navigation system. Okay, so so for a lot of people, you can ask anyone, they'll all probably give you a different definition of what it is to them. But essentially, it's made up of 5 primary components, okay? We'll just skip to the little graphic as well. So the formal definition is GIS is a computer system for capturing, storing, checking and displaying data related to the Earth's, to a position on the Earth. And that's the most important part here. It's spatially enabled, okay? So everything happens somewhere. And GIS in itself, we've got hardware, which would be your navigation devices, your satellites, your GPSs. Okay, we've got software, you know, you've got your QGIS, your ArcGIS, and a whole bunch of other different types of software. We've got apps. GIS is moving into the app world, so you're able to download apps on your phone, and these things are spatially enabled. We've got data, obviously, data collection, data repositories, all different types of data that has a location associated to it. And then, of course, there's the people, there's the users of all of these things, okay? And then also the people that make it work and make it happen. So, getting here. As I said, everything happens somewhere. It happens at the point in space and at a point in time. And with that, GIS can help us to identify problems, we can monitor change, we can manage and respond to events, we can perform forecasting, we understand trends, whether it's spatially related or if they are related to each other in any kind of way. And we can also set priorities. Okay, so there's a lot of different ways that GIS can be applied. And of course, again, it is relative to your specific context. So in GIS, we work with different data types, okay? And very basically, your primary sets are raster types, which is imagery, okay? So think of your aerial photos, your satellite imagery, etc. And then there are your vectors, which are points, lines, and polygons, okay? Your data, if we have a look at on the side here, this is just an example. So we have the real world, and the real world can be split up into these different data layers. So we'll have a raster image, which many people just use as a backdrop. We can have elevation models applied to that, transportation routes, such as your road networks, your railways, etc., addresses, your point locations, your property boundaries, water features, survey control. All of these data can all be ingested into GIS, obviously providing that it is in the appropriate format. Okay, so in the system itself, we'll import these layers and they are stacked on top of each other to create a representation of reality. Okay, and then we can use different types of analysis tools to evaluate the relationship between these layers. Okay. Spatial, it can be spatial relationships. It could really be anything. We've a lot of analyses use hotspots, you know, as density maps. The possibilities are really quite endless. Okay, and normally with GIS, the output is a map. Okay, and these maps are used in policy context, they're used for forecasting, they're used for planning, mitigation, response. Any type of decision-making process can benefit from a map as well. Okay, and the data and the potential of GIS is really just limited by the user. And then of course we have data quality aspects as well. We know, or we have a common saying, garbage in, garbage out. Okay, so that's one of the things I want to touch on when I get down to the data. We're looking at data quality. If your data quality is not good, you cannot expect to have a great result or accurate output. Okay. So in terms of software, you get proprietary software, which would be your paid software licenses, such as ArcGIS. Many people will be familiar with it, as many organizations across the world use it. Okay, but then you also get your free and your open source ones, which would, a good example is QGIS. There is no real better one. The best GIS software is the one that works for you and the one that you are able to use. Okay, so your need, you know, don't spend a lot of money on software where you're just going to use it for very basic functions. Okay, you need to, obviously, if you do not have GIS and you want to go and evaluate this, consider what it is that you need it to do. And there's a lot of examples on YouTube, on the internet, etc., where you can go and have a look and assess these. You can even get demo licenses of the proprietary brands where you're able to test these out for yourself. Okay, and then finally, with all computer software, you have to be mindful of your computer's specifications. You know, your hardware, your RAM, how much do you have a graphics card, etc. And obviously, it all depends on the analysis you want to do. But you know, don't spend a lot of money on software that you can't even install on your computer. Okay. Okay, so now we're getting closer to how we're going to be using GIS in the context of benthic sampling. And I think it's important to just touch on methods and standardization. So your methodology, that is basically how your data was collected or generated and how it was analyzed, okay? Your standardization would be creating or implementing norms, so keeping things the same. So if we're using meters rather than kilometers, you know, we all sticking to the same thing. Okay, so that's an example of standardization. Your methods in GIS, when documenting it, it applies to GIS exactly the same as it does to any other research. Okay, and what we really want to do is to have well-documented methods to ensure that, firstly, you create a sense of transparency so everybody can see how you derive your results. And people can trust it. People can also replicate it. So if somebody is interested in your methods and they want to apply it to their little study site, they have a clear guide as to how to get that right. You can think of it in the context of having a recipe and you want to bake a cake or something. You know, it has to be quite exact if you want to end up with a product that looks like the picture. It's exactly the same in GIS. Okay, so with that, reinforcing consistency and uniformity. And as I mentioned before, it allows others to repeat the process. Okay. So in the context of benthic sampling, we've got some primary steps that we had to follow in order to design our methods. Okay, so the very first part of it is defining the study site. Okay, we'll go through each one of these in detail. I'm just giving an overview at the moment. The next component is getting an understanding of what is in that area. You've chosen a site, so what is it that we can expect to find there? And they will draw on different types of data sets, you know, just to give us a more clear understanding. This type of assessment is a desktop assessment, so it does not replace an actual field site inspection, but it does give you some idea of what you can expect prior to venturing out. Okay, we create our sampling grids. We'll select the sampling stations, and for that there's two methods, a manual method or a randomly assigned method. I will go through that as well. and then recording your findings. So first step, defining your study site. As you can see in the map here, we've got 6 study sites that were selected. For the purpose of this presentation, I will only focus on one, but I just wanted to give you a broader context. So this is Cape Town. So this is Cape Town in South Africa. We've got the Kabonkelberg, Olifantsbos, Cape Point, Paulsburg, Castle Rock, and Boulders. Okay, so those were our study sites that we had selected. And at this point, we don't actually know much about what's going on there. So in order for us to get an understanding of the area, we were interested in the substrate, the bathymetry, and just some information regarding the protections of the protected area zonation. So for Kabonkelberg, you can see that there is a distinct line over here, and that speaks to the zonation. So everything within this yellow block is within the marine protected area. The zonation is how the marine protected area is basically divided up. So this is a restricted zone with the red lines, and this is a controlled zone. And you see that in the Kabonkelberg area, there is a distinct divide. So part of our study site here falls within the restricted and part of it falls without. Similarly, we see this here at Olifantsbos and also at Paulsburg. Yeah, just for this purpose, I didn't include the other study sites. We also then looked at the geology. So we're looking at substrate. So that's important because of the type of sampling we're going to be doing. And I think one of the other presenters will touch on that in a bit more detail, but we were interested in rock or hard substrate. So we can see there in the Kabonkelberg, the dotted area, we can see that there is rock substrate there. Same with Olifantsbos. Paulsburg, however, just has rock substrate as we go closer to the shore. Okay, so this already tells us a bit about what we can expect in the different study sites. Okay, and then of course we know that they all fall within marine protected areas. And we can also use this information then to eliminate things we don't want. So for example, if we want to focus solely on rock substrate, then perhaps Paulsburg might not be the best location to go and do our sampling, whereas Olifantsbos and Kabonkelberg seemingly occur on rocky substrate. Okay, so now that we've got our study sites, I'm going to focus on the Kabonkelberg one. We are going to create our sampling grids. So the software I work in is called ArcGIS. So that is a proprietary software. And that's just because my employer, we have licenses assigned to us. And yeah, everybody uses it. So it's very easy to exchange data with others because we're all working in the same format. So with the sampling grids, we do have some considerations. Okay, so do we want to define the number of grid cells? Okay, that's the number of squares or rectangles, or or how we're going to divide up our study area. We can do that by either defining the numbers. So if we want 10 grid cells across the whole study site, we can tell the system to do that. Or do we want the cells all to be a specific size? Okay, so this is where you define the exact size of each cell. And in our case, we wanted each cell to be 250 by 250 meters. So those are squares, obviously. Okay, and in ArcGIS, we use a fishnet function. Okay, but in other software, they would also have a similar tool. It might not be called fishnet, but there are definitely tools available in other softwares in order to be able to generate grids. And the nice thing is that the data format in itself is compatible between different softwares. So I can take a file that was generated in ArcGIS, which would be a shape file, and that is ArcGIS proprietary format. And I can actually open it up in a open source system such as QGIS, because it would accept shapefiles. So just to illustrate here with these two arrows, we, as I mentioned, we wanted it 250 by 250 meters. And this is what the tool looks like in ArcGIS. And here we just specify the cell width, 250, and the cell height to 250. Of course, if we wanted to go by defining the number of grid cells, that we would then enter that information here. How many rows do we want? How many columns do we want? And whatever you tell the system to do, it will try and do a best fit. Okay. So once we've run that, you can see that we'll get our grid. Okay. So this is obviously land over here. So we eliminate cells that do occur on the land, if that's the case. But yeah, we can see the rock substrate below it, and it seems that the whole study site does occur on rock substrate, which is exactly what we wanted. We can zoom in, and if we wanted to, we would be able to delete these areas here, these cells that occur partially on land, or maybe this area is too shallow or so. But GIS in itself gives you a lot of control over the data. So you're able to eliminate cells that you don't want if you no longer need it to form part of your analysis or part of your study site. And then combining data sets, the blue at the bottom, we obviously know it's the ocean, it's the seawater, but it also contains depth information. So we have a very, very high resolution grid for the bathymetry. We got special permission to utilize this information. So you also need to be checking with your local agencies, like your Navy or other units involved in marine activity, who maybe does bathymetry scans, etc. So with that, we're able to just indicate what the average depth of each grid cell is. Okay, so these numbers on the top here, those ones there, 99, 100, 101, 102, etc. That is just the number of the grid cell. So attached to this in the back end is a table. Okay, and that table will enable you to select the different grid cells. And yes, so that one there is just your grid number, just so that it has a reference. But this bottom number here, that is our depth. So when we click on that, we are pulling through the average depth that occurs within that 250 by 250 meter grid cell. Okay, and with that, you're also then able to make further decisions about whether this is too deep for your sampling, or maybe it's too shallow for your sampling. But yeah, it's just to further illustrate the use of different types of data and how it can inform your sampling. And again, this was just using the zonations information. So we see that half the site, or not even half the site, most of the site here occurs within the control zone, and the ones closer to the shore occur in the restricted zone. Okay, and with this, you see, because we've got the grid numbers, we're able to tabulate. We can add fields to the actual data, increase the information in the tables to say that, cell 199, cell 200, etc. They all fall within, or predominantly fall within restricted zone. So we can use all of this information to enrich our data. Okay, and then the next step would be, now that we have our grid and we've got some awareness of the area in terms of the depth, control zone, restricted zone, etc., we can now go on to assigning our sampling stations. Okay, So this can be done manually. Of course, you can always go to your cells and just choose them as you see fit. But I want to illustrate another tool from ArcGIS. Again, this is also available in other software, but it just creates random points within a defined area. So you have to tell it what to do. And in this case, I told it I want 18 points, and these points must be at least 300 meters apart, okay? And the reason I chose 300 meters is because, remember, we said that the grid cells are 250 by 250 meters each. So we want points, we don't want points to occur in the same grid cell. Okay, so if we had to choose 50 meters, we might end up with 2 or even 3 points within that same grid cell. So that we specify over here. So we see the number of points, and I said I want 18 points. And down here, the minimum allowed distance between the points. So if you have a single point, it will consider the radius. So it's considering a 300-meter radius around that point, and it will not put another point there. Okay. So we run this tool. Once we filled in all the parameters, etc., we'll press that OK button and it will run the tool. And that is what we end up with. Okay, so I just made a label there, random points, and I made them as triangles. So you can see that these points are randomly generated, and you can run this tool as many times as you want. If you're happy with the way they occur here, or you think that maybe there's too many points happening here and not enough on this side, you can move the points around. You can do whatever you want to, but this is just a way of generating random points in a spatial context. And then, of course, we can further interrogate where these points have fallen. So if we didn't eliminate cells in the previous round when we generated the grid and we considered the depth, we can do that now. Maybe we've got too many points occurring around the 95 meters or 90 meters or so, and we want it to be a bit more diverse. Then we'll just rerun the tool, or we'll just manually move points over. Okay, so now that we have created our sampling grids, we've selected our sampling stations, I just want to speak a bit about recording your findings. Okay, so as I've said, GIS has the ability to store attribute information. So all those dots in the maps, all the grids, everything you see has a table attached to it at the back. Okay, so just as an example, I'm considering the controlled and restricted zones again. I'll just show you what that table looks like. So we've got our different, we had our different study sites, and we see that we've got restricted zone there, and that one as well. So restricted, restricted, restricted. There's a controlled zone over there. So this is just ways, again, how you can enrich your data. So you don't, so literally when you have everything in, you can click on it and it will give you this information. Okay. So now we're moving on to a bit more advanced techniques. The methodology that was just described was quite simple. Okay. So in GIS, when we have a method that we repeat a lot, we like to develop models for that, and it's not as super complicated as it might sound. It is considered an advanced GIS technique though, okay, but we use it to automate processes and limit the button clicking. Okay, so consider if you're getting a volunteer or intern to assist you, and you just don't have the time to explain to them how to develop this grid. And then generate the random sampling points. You can take all of those processes and combine it into a model, okay? You still have to provide the model with the instructions. For example, how big do you want the grid size to be, the width, the height, etc. But it will just be a matter of typing in those parameters and then clicking the OK button, and it will then go through all the steps that we just went through now, that we done manually. It will go through it automatically and generate your grid at the end, as well as your random sampling points. So it does save a lot of time, especially when we do repetitive tasks. Okay, and then you can build standards into your model. So tell it that all outputs must be in meters, for example. It also has the potential to minimize the human error that could be introduced, because you might be a lot more familiar with your technique than somebody else, and they might just click the wrong button or something. So yes, so models do help to speed up processes and eliminate the potential for human error. And these pictures at the bottom here are they're just illustrating different models. So this was just through a quick Google search of models. All of the yellow ones here are the actual processes. Blue is input data. P, that means it's a model parameter. So when you when you start up this model, you have to specify these parameters. Okay, the greens are just the outputs. Okay, and why this one here is a model parameter as well is because we want, this was just the ability to rename the output. So you see in this one, and I mean, don't worry that we can't see the actual labels. We just see that these rectangular orange yellow blocks are all the different processes. So some processes are reliant on others, and we first have to generate some data before others. It can get extremely complex, but again, you can pass on this model to the next person who will then just be able to put it into their GIS system and run it. And it saves on a lot of time and a lot of hassle. Okay, so if we consider everything we've done with the benthic sampling design, with benthic sample design, sorry, how is it that we can proceed to then create a GIS model for this? Okay, and it's really just considering all the previous steps, and isolating the GIS components. Of course, the non-GIS component, I would say, is where you're taking the different data sources, like your rocky substrate. Well, even that can be automated and included in the model. But if you want to make a judgment call about depth, for example, or you want to see control versus restricted, while we can build these into a model, we don't always want the model to just make decisions for us. So for the purpose of this exercise, we'll just isolate the core GIS processes, and then we'll use Model Builder. That's what it's called in ArcGIS, but of course QGIS also has one. And we'll sequentially construct the steps within that model. Okay, and we'll assign parameter fields as well, because these are the ones that are going to rely on user input. So if we essentially simplify the entire process, we would be defining our study site. So we'd be delineating the polygons, and it tells the model only perform the calculations in this area. Okay. We're defining our grids. So we can do that by the grid cell sizes or the number of grid cells that we want. And we can use the fishnet tool to create that grid. And then finally, as the last core GIS process that we'll rely on, is to define your sampling stations. So if you're going to do a manual selection, then obviously this one would not apply. But otherwise, we'll go with the random points generator. We'll tell it the number of stations we want, and we'll tell it the minimum distance between the stations. And we put these, as we see it displayed here, we'll put that we'll construct the model, and then it will provide us with the output. Okay. So as we move towards the end of this presentation, I just want to touch on data considerations, because I showed you some really great data sets that we use, especially the bathymetry ones. The high resolution bathymetry is usually the difficult one to come by. Okay, but still, we don't blindly trust data. Data is a representation of a point and space in time. Okay, so it's what the state was when that data was generated. Okay, and anything could have happened in that time. Things could have changed. Yeah, so we always have to, it seems like a no-brainer, but we should look at, timestamps of data. Is there update? When is the data relevant to? Okay. And then just more broadly, in terms of GIS data, we have to consider the spatial scale and spatial errors. I have the picture of a bunch of GPS and navigation type devices here, and they all have different accuracy levels. Okay, so really also you need to consider the accuracy that you are interested in. Incomplete data, maybe using somebody's data that where they haven't captured everything or there are fields missing, etc. Temporal issues, that's exactly what I just spoke about, the timestamp. Are you using a data set that was generated in 2002, and it's almost 20 years old. So we have to be mindful of that. The attributes, incomplete or erroneous attributes, that's the data that you find in the tables that are attached to the spatial component. Okay, so those might also become outdated or change. A very important one here is scale and resolution. And I say that because sometimes data is just very scarce. And we want to find a means to substitute that, to fill that gap. So just be mindful of global data sets. And why I say that is because, especially with bathymetry, many of us use the GEPCO bathymetry, but the resolution is often too low to really be useful especially in this kind of context. Okay, so just very simply put, if the resolution is 1 kilometer, for example, you cannot use it if your grids are 250 meters. So if the resolution is bigger than your grid cell size, it's not going to be as useful to you. It might give you a good indication or a fair indication of your underlying bathymetry, but the detail that you saw in the maps that I had, you will not be able to achieve that using a coarser resolution grid. And there is a link over here, and it does speak to those interested in flood modeling, but the principle is the same. Okay, and just to wrap up on my last slide, some tips for just regarding data, maybe not necessarily GIS, but data is an asset and its value increases with age. And we're seeing this now, especially with all the climate change studies, people turning to historical data to see trends and then do forecasts and projections and all these things. So the value of data is truly being recognized. And I always promote, consider depositing your data in a repository. Okay, just simply because we don't, hardware can be quite unreliable. You can't anticipate your hard drive crashing, your computer being lost or damaged or anything. And the data that's on there, once you lose it, it might be gone because you'll never be able to go back in time and resample, at that specific or get data that's specific to that that point in time again. Okay. Repositories are also designed to protect your data, so there are backups and fail safes and all these things. And if anything does happen to your hardware, your PC, your laptop, your hard drive, you'll be able to recover it from somewhere. Okay. And then repositories also make your work visible. So maybe there's somebody on the other side of the world doing similar types of sampling, and they can draw lessons from the data you've collected. Yeah, so it makes it searchable, discoverable. You can also get DOIs, and people who use your data would then have to cite your data as a source. Okay. Importantly, data does not have to be perfect. So we encourage people, if you have raw data, to put your data in a repository. You can publish raw data. Okay, and if you do some processing or anything on it, you know, you can update that with a newer version, because not everyone actually wants to use processed data. Some people want to use raw data. Okay, and then also when you do publish data, you have to list the limitations. Okay, and also speak about the formats and the appropriateness of the data, because nobody would know the data better than you. And with that, I thank you for your attention, and I hope you found it useful. Thank you.
Great. Thanks for the very nice talk, Lauren. I think, is Lauren live? To be able to take any questions? Yes. So the first question is, what is the main challenge you encounter related to making your data available, and what repositories are dedicated for that in the WIOMSA region?
Hi, thank you. The main challenges with making my data available, well, I actually work in an environment, and it's quite an enabling environment, because we have an internal, a dedicated data repository. And that data repository is called the Marine Information Management System. So in terms of depositing data there, we're still in quite the early stages, where we are encouraging all of our scientists who collect data to store their data there. But yeah, I think before that repository existed, it was really just finding that home. Where, you know, where can I actually put this that it would make sense? But I work primarily in a GIS context, and with GIS and spatial data, and there isn't exactly a dedicated repository for that. So we need to take advantage of these overarching repositories that are available to us, and, you know, realize that even if you don't just, you know, you don't collect specific biological data or oceanography data, your data still has relevance and can be put in those types of spaces. So even though your data is different, you shouldn't be shy about putting it, or even just asking to put it somewhere. Yeah, and more than often, these people that work in these repository environments will be able to guide you, you know, if your data is maybe not suited for that space. I know that WIOMSA had a clearinghouse, but I don't think that's a very active space at the moment. And I think there was at some point a large focus on having GIS data stored there. But for me personally, I utilize our in-house repository for data storage. Yeah.
I guess there is a related question to that. That you mentioned already that some of the data don't have to be the processed ones, the ones that you end up depositing in the repositories, but there might be also raw data as well. But there might be also specific raw data, for example, that might be sensitive, that you maybe don't want to put out there. So for these types of data, is there sort of, what is the best way of, I'm guessing the question is, if you want to store those data, one of the difficulties is it might be expensive, for example. So do you know of any sort of cheap but secure at the same time options of where you could be storing data? I guess this does not only apply for GIS, it applies for data management in general.
Yeah, yeah, that's actually a very very popular question. You know, I have data that I want to publish myself. I don't want anyone else to use it yet until I've published. So what do I actually do with it? And in our system, we make provision for embargoes to be placed on data. So most repositories will have a data policy associated with it. And that embargo would usually, they'll specify the time period. What we aim to do here is for the data owner to publish their metadata record. And that metadata record is what will be available for everyone to see. And metadata is just, it's data about your data. So where did you capture the data? Where, you know, what format is it in? What are the variables you collected, etc. Just to make sure that everybody out there knows exactly what type of work is being done. So there's a secure space that we have that where we'll store that embargo data. And if anybody is very interested in it and wants to have a discussion about it, we'll refer them directly to the data owner. So in that way, the data is still safe and backed up and secured in a repository, but nobody else has access to it. They only the data owner, and the data owner is the one that will grant you permission to utilize the data, or even just to view a sample of the data. Yeah.
Yeah, great, thank you. Yeah, that's important information, because a lot of the data that is being collected is often not necessarily for public consumption. So there is another question from Liban, which says, thanks for the informative presentation. Do you think it would be more understandable if you could include live examples of using the application? For example, how do you input grids in your sampling area? I'm guessing maybe it sort of relates to if in order for someone to sort of like understand more like some of these examples, maybe there are any sort of resources available online that might sort of someone might be able to use.
Okay, sure. I mean, a lot of these tools are, you have little demo videos and tutorials on YouTube, etc. So for example, to create the grids, I use the fishnet tool in ArcGIS. And that fishnet tool is not specific for benthic sampling. It can be used in any type of environment, really. In a previous life, I was doing some household surveys, etc. And we also had to create random points for households that we were going to survey. And we use the same fishnet tool. So I think it's just a means of, if you are interested in that, in the method that I used, to just go and Google the ArcGIS fishnet tool, or even just fishnet tool, and it maybe can provide you with examples from other softwares as well in order to generate those grids. So yeah,
a lot of that information is available online, essentially.
Absolutely, yes.
So I had a question of myself. So I think you mentioned during your talk that the resolution of your data, for example, bathymetry, might be not as good if, for example, you're using a global data set. How do you know it when you start using a data set in GIS? How do you know the resolution of that data set specific to your area?
Yeah, so it's actually GIS standard that you have to provide metadata, the metadata file with your data. So okay. in some repositories, it just appears on the web page prior to download, but in other repositories, you'll actually get a PDF document or something as part of the package when you do download. And in that metadata, as I mentioned before, it describes your data. So it will tell you the resolution of it. So you know whether it's going to be worth your time to download it or not. Yeah.
Okay. So that's an important thing to check then before you actually start using a data set to see if it's relevant in your study.
Yeah. Well, it will at least save you the time of downloading it and discovering it later. But yeah.
And a final question, I don't see any other question. I have another question for you. Is I think you mentioned that in many cases it helps a lot if you sort of liaise with agencies, for example, national agencies, yeah, if they have any sort of data. So from your experience, is it usually data that is easily accessible if you do sort of like monitoring work, for example?
Some data is more accessible than others. We do have a national initiative for all data sets held by government institutions and agencies to be made known. So more and more, we are having institutions have a list of the available data and on what people have actual access to, which is a really great initiative, because even if the data is maybe not fully publicly accessible, for example, the high resolution bathymetry, they wouldn't give me the actual site that I was requesting. They gave me a sub-sample, sort of just enough of the area that I needed in order for me to do my work, because there were some sensitivities around the data. But I mean, you wouldn't know that unless you go knocking on people's doors and start having this conversation about data availability. So we do actually have established national forums where people come together and we literally talk about data, data availability, data access constraints, etc. Yeah.
See, well, thank you. I don't think we have any other questions. So we'd like to thank you again for a very nice presentation.
Thank you.
We're now going to move to the next presentation by Dr. Tanja Haupt. So Tanya is a marine researcher at the Department of Forestry, Fisheries and the Environment. Her work involves surveying, monitoring, as well as examining the effects of environmental change on benthic invertebrate communities. She's coordinating a shallow water benthic monitoring program, which is targeting marine protected areas, as well as a conservation physiology program where experimental biology and physiology is being used to examine how various environmental stressors like warming or ocean acidification may affect seabed selected benthic taxa. So in this talk, she's going to be talking about exploring, monitoring benthic biodiversity and local lessons and a note on sampling design. So again, the virtual floor is yours, Tanya.
Hi everyone, I'm Tanya Haupt, and my talk will focus on some of the projects that our Benthic team at the DFFE, based in Cape Town, South Africa, have undertaken or established. Most of the examples are from within our local priority areas, each with a unique sample design. A brief outline of the talk. Firstly, why is it important to monitor biodiversity and benthic invertebrate marine communities in particular? I'll then touch on why it is important to have monitoring programs in priority areas such as MPAs, marine protected areas, or IPSAs, also known as ecologically and biologically significant areas. And lastly, I'll show you some projects where we've explored benthic invertebrate communities or managed to establish monitoring programs using different sample designs. Why monitor biodiversity? When we have an ecosystem that's in trouble, maybe because of changes in environmental variability, an oil spill, alien invasion, or even overexploitation, the most essential tool to have is knowledge of what that ecosystem was in its pristine state. This highlights the importance of having baseline biodiversity data so that we actually have something with which to measure the environmental impacts caused by various factors. And this is why currently it is the mandate of various organizations to monitor biodiversity, including that of the DFFE. We have various programs that monitor invertebrate biodiversity in different ecosystem types within our department. For example, rocky shores or estuaries. For the rest of this presentation, I will focus on benthic marine communities. The benthic organisms we target for monitoring are mainly sessile, meaning that they do not move. These are your sponges, your coral, bryozoans. So why study or monitor these communities? First, besides the fact that they are so diverse and fascinating to explore, they also include largely unexplored key ecological species. Benthic monitoring is also a relatively effective and reliable technique that can detect subtle changes that serve as an early indicator before more drastic environmental changes occur. They are also especially effective in assessing long-term changes by responding to stresses like ocean acidification or warming through detectable changes within populations over weeks and years. This is in contrast to plankton, for example, where populations show a more immediate change with no long-term consequences. And because they are sessile and don't move, they must either tolerate, adapt, or die. What is the importance of conducting such research in priority areas such as MPAs or IPSAs? Many of the existing MPAs in South Africa were established with a strong focus on fisheries management or the conservation of species of high economic value. As such, in the past, there was a general disregard for non-commercial or non-focal species, which may lead to a poor scientific base or understanding of whole ecosystems. Thus, establishing long-term monitoring programs would not only provide essential baseline data, but also a better description of and understanding of how these communities will change over time. Information of this nature can only strengthen conservation decisions. One of the main outcomes of many MPAs is that they have been established to conserve biodiversity. And I put these in inverted commas because how do you conserve what you don't know is there? With long-term monitoring programs, we will have a better understanding of the full range of organisms or whole ecosystems that priority areas aim to conserve or already are conserving. All useful information when establishing and managing these areas. So now that we know more of what benthic organisms are and why they're important, particularly for monitoring purposes, I will take you through some of the projects that we have undertaken or have established within our local priority areas for exploring and monitoring benthic communities using different sample designs on both a large and a small or fine scale. I'll also touch on how these designs differ from that of the past WIO expeditions. We know that sample design will vary depending on your research objectives. I find that research objectives are often also based on what you and your team are capable of doing with the infrastructure or human capacity that you have available. For example, do you have standard versus more advanced research tools? Do you have small versus larger research vessels or staff with specialized skill sets such as skippers, divers, dive supervisors, or even pilots that are licensed to operate some underwater camera systems? And when comparing our projects and sample designs, the one thing that we probably all agree on as a team is the advantages of using remote underwater camera systems. Not only can we sample to greater depths, but we can also easily standardize protocols between sites and areas. And we can now also conduct extensive sampling of benthos in even the smallest conservation areas where traditional methods of sampling can be really destructive. Of course, although we recognize the utility and importance of this methodology for research, we also recognize the importance of reference collections for taxonomic verification of the benthos that we observe in our images or videos. However, coupled with benthic camera systems, this physical sampling can certainly now be minimized. An example of sampling benthic communities on a large scale would be the benthic surveys we conducted off the RV Algoa along the southernmost portion of the Benguela ecoregion. The overall objective was to provide a better understanding of the benthic invertebrate communities and habitats in high priority and adjacent areas, such as the Robben Island MPA, the Table Mountain National Park MPA, and a section of the newly proposed Cape Point to Cape Agulhas Epsom. For the sample design, a grid was placed over the sampling area And sampling points, what we call stations, and these are the yellow and red squares, were then chosen by first dividing the grid up into varying depth bins, and then randomly selecting a number of stations within each depth bin or depth category. In this case, we had 5 depth categories between 20 and 250 meters and randomly selected 10 stations in each. Since the Robben Island MPA was only recently gazetted, a set of stations unique to this area was also sampled to gain a good representation of the benthos and associated habitats. Data collection were via visual surveys from benthic camera systems and also physical samples that we collected from outside of the MPAs, like dredge or grab specimens and also geological substrate. Oceanographic or environmental data was also collected. For example, temperature, amongst other variables, which may very well be contributing factors influencing the distribution and abundance of benthos within this area. This extensive assessment of the benthic communities in the southern Bankwala region allows for long-term monitoring and may be repeated every 5 or more years. Focusing on Table Mountain National Park MPA in particular, we have an example of monitoring on a smaller scale. Here, the objective is to conduct long-term monitoring of benthos with a particular focus, on rocky or reef habitats. As for the previous project, we work closely with our GIS specialists to place monitoring grids over areas or sites of interest within the MPA. To date, we have initiated monitoring in the grid furthest north. This is a large stretch of granite rock extending to 100 meters, part of which falls within a restricted or no-take zone of the MPA. That is the Kobonkoberg restricted zone. And we have also identified areas further south and in the False Bay restricted zones as hotspots for benthic and fish diversity. A closer look at one of the grids. Seen here is the grid north of the MPA, part of which falls within the Kobonkoberg restricted zone. This grid will be sampled biannually at the beginning and end of each year, and for each sampling trip, we randomly select 20 stations that fall within varying depth bins. In this case, the number of stations differ per depth bin, since the grid is made up of mostly deeper stations that lie between 75 and 100 meters. They are therefore more deeper than shallower stations. Now, these are smaller grids or areas that we target compared to the previous project or design, and the shallower sampling depths are at 15 meters and therefore easily accessed via small boats rather than a big ship. Data collected are visuals or photo quadrats by a drop camera system, which is a simple locally built construction, more of which you will learn about in tomorrow's session. We also attach a little temperature logger to collect environmental data. Seen here is an example of some of the images of benthic organisms that we've obtained so far from 15 to 100 meter depths. Another example of monitoring on an even finer scale is along permanent or fixed transect lines at diving depths of less than 30 meters. Again, we target rocky substrate, and the objective is to monitor the sessile benthic communities that occur along these fixed transect lines over time. We have 2 such sites within Table Mountain National Park, NPA, and this diver swim map shows one site where we've installed pins as marker points. So the idea is to conduct photographic dive surveys all along these lines. This slide shows the process of installing the pins, but even after ensuring that we had recorded their positions as accurately as possible, we have encountered so many difficulties in locating them again. This, coupled with waiting for good weather days and recruiting a suitable team consisting of a skipper, divers that are familiar with the area and the location of the pins, a dive supervisor, has resulted in this monitoring attempt not being nearly as successful as the projects in which we used remote underwater camera systems. And then lastly, a very different sampling design seen in the past WIO2 expeditions of 2017 and 2018, where sampling extended from Mozambique to Tanzania and all the way to the Comoros. Instead of grids, stations were randomly selected at varying depth bins, shallow and deep, along predetermined transect lines. Some of these lines were already in existence, such as the Nansen or other identifier priority lines of national importance, and some were added specifically for the WIO2 expeditions. In this case, because the approach was multidisciplinary and the focus was not on collecting benthic biodiversity alone, but also other disciplines like oceanography, we needed to compromise and could not always sample in the habitats or areas of our choice. A consequence of this, the habitats in which we sampled were extremely silty and muddy. But despite this, we have a really good collection of samples, images, and video footage. Thank you for listening, everyone.
Thanks for the very nice talk, Tanya. I think Tanya is live and she can join us for the Q&A session. I have a question for you. I think you mentioned that project around Robben Island where you were monitoring it. I think I saw it was extending to depths up to like 200, 300 meters. I'm not really familiar with the location, but that sort of include what we call mesophotical ecosystems, which you have usually between 30 and 150 meters.
Yes, yeah. So basically our shallowest depths were at, say, 16 meters. So it's extended from 16 to 250 meters. I think Robben Island wasn't as deep. But yeah, I mean, the differences in community assemblages between depth zones were, I mean, they were quite distinct, even in the Robben Island NPA. We saw also a lot of rock lobster assemblages, and although we mainly monitor sessile inverts, that was one of our benthic findings that kind of appear in almost all our
And I guess because a lot of the information from these ecosystems, which are not studied that much, is not out publicly available. Can you hear me?
Yes.
Yeah. So what I wanted to ask is some of that information from that monitoring program, is it available online or is it sort of still being prepared?
Oh, okay. So basically, we started with our monitoring in February of this year. So it's quite new program. We actually plan to sample more of our grids now coming in 2 weeks in September. But the plans of the data at the moment, it's not publicly available. But we do plan to put it into or deposit it into our MIMS repository system. So yeah, and that would be available for people to use as they like.
Yeah, it would be great if you can add a link later on about that repository,
yes.
Because I'm working like on these ecosystems and it's really difficult to find information from other locations. So if that's available publicly, then that's great.
Yes.
We do have a question from Jamie Caesar, and they're saying, hi Tanya, you said you randomly selected the sampling grids. Doesn't this run the risk of missing some grids and oversampling some others? How do you circumvent this?
So because it's a random selection and because it's a long-term monitoring, the chances of us every year we randomly select different cells. So there is a chance of us selecting the same grids, same stations. But yeah, over the years and as time goes by, we actually do get to most of the cells or stations within one grid. And sorry, Paris, coming back, yes. So I think I understand better now your first question. The data for the Robben Island MPA is actually part of an MTech study. So that data is actually already in the repository system. But one of our technicians is currently busy writing it up as part of his MSc. So once that is done, it will also be in a nice paper format, hopefully.
Yeah. Are you planning on having some sort of field ID guide, like the one for the invertebrates that was published a couple of years ago for South Africa?
That would be cool.
I don't know.
Nice idea.
Really useful, yeah.
Yes, definitely.
So there is another question from Roman. They're saying, over the period of a survey, have you found any impact of temperature rise on the species? And how is the recovery in those sites that you have monitored, for example?
Okay, so as I'm saying, so the first studies or the first surveys we only managed to do in February of this year. We plan to sample more now in September. So that whether there's any temporal changes in temperature and the effects thereof, because it's a long-term study and it's designed in that way, we will only be able to answer that in a few years' time. So the more we sample within the study area, the better, and the more data we collect. Yep.
Another question from Ludovic, I think I'm pronouncing it correctly. Very nice presentation, thank you. Did you find any particular habitat or unexpected benthic species during your sampling, particularly in the mesophotic zone where there is limited information on those ecosystems?
It depends on what study or what project they're referring to.
Well, I'm guessing as a general sort of rule, did you find anything that you weren't expecting to?
No, I mean, we've worked in this area for quite a while now, so we haven't found, I mean, we find all our species or our organisms that we find interesting. We do have Lorraine, like I was saying, that is looking at each image or each video that we have of this area in detail. So he might be able to shed more light on if there's any particularly interesting species. But yeah, for us, for example, for our Kobonkelberg grid, just some preliminary analysis, we find that there's definitely distinct community assemblages between the depth zones. So we find that the deeper zones, for example, they have a greater variety of cnidarians or bryozoans or corals and yeah, so for us, that's we're not looking at species or specific organisms, but kind of, you know, broad benthic taxa. So, and across the depth zones.
Yeah, yeah, understood. There is another question from Kelvin. They're saying, if you use camera photos for sampling, don't you face the risk of missing some of the species? I guess this is a big probably theme running through the workshop. And what do you do if you need to collect some samples on reef or rock shores?
Yeah, yeah. So, I mean, that is the risk that you take when you do benthic sampling via remote camera systems. But what we do also try to do is sample particular areas to get some reflections. So, for example, with our Southern Bangalore project, we would do a few dredges outside of the NPA. So if we saw an image inside the NPA with our camera systems, we could kind of, how do you say, corroborate the data or validate the data with those physical samples we collect without kind of destructive sampling outside of. Yeah. And I mean, people have been working in Table Mountain National Park in PA for quite some time. So we do also have some physical samples.
Okay. There is another question by Saeed, and they're saying, what happens to fauna organisms? Do you collect samples from those as well? So things living in the sediment. If you collect samples from those, yeah.
But one of our cruises that we went, that we did back in 2019, we have our, it depends on who we have on board at that time. For example, we invited our geologists with, and they took some grab samples. So we don't look at it specifically, but we do give it to institutes that are interested in that sort of thing. So we basically just focus on your macro benthos, so the things that you find on top of the substrate.
Okay. Two more questions before we wrap up. One by Frederick. So trawlers are the real threat for benthic ecosystems. To what extent do you think that trawling data is imperative in order to study benthic communities? And if you have been using, for example, trawling data to inform your benthic community assessment.
What was considered? The data from trawlers can be used to study to what extent do you think? Sorry, I'm just reading it out.
Yeah, that's fine.
Like I was saying, with our dredges as well, I mean, you do also need your physical samples. So, but at least when you have camera systems involved, that type of destructive sampling can certainly be minimized. Yeah, so for example, instead of trawling at every, say you have your grading, instead of trawling at every station, you might select a few stations where you can trawl or dredge. And, you know, most stations within whatever area you're working, you can kind of use benthic camera systems. So no means are we saying that benthic camera systems, you know, are take the place of other type of sampling, which is saying that it does have its advantages. It is non-destructive. It does allow us to go to deeper depths than we could previously.
And final questions, because we don't have any more time, is it possible to carry out this methodology but with a small boat of 6 to 9 meters? So some of the sampling that you have done, for example?
Yes, definitely. So our drop camera surveys that we do in Table Mountain National Park, so our more fine scaling monitoring that we do off a rubber boat. So we don't have a big ship for that. yeah, and I mean, for us, because it's in our local MPA, it's day trips. You know, we don't get on any fancy boat. We just go as a team of 3 or 4 people, get on our little rubber duck with our little drop camera system. We do our sampling. Yeah. And I think for a lot of countries, that would probably be the most feasible or the most likely way that you would get to sample Benthic communities using underwater camera systems, like if you had a little drop camera.
Great, thanks. I think there are 2 more questions, but because we've just sort of reached the end of this session, I don't know if you're happy to sort of like answer them now, or do you want to answer them later via email maybe? Or...
I can answer it quickly. There's one that asks whether we do the sampling at midnight. So no, I mean, our data is not really dependent on any, time of the day. So no, to answer your question, I mean, if you want to go, we don't. I think there's safety concerns for that. And the data collected in Mozambique can be shared with us and also involved. So at the moment, the data collected on the WIO2 expeditions belong to the countries of Mozambique, Comoros, South Africa, and Tanzania. So Stella, you could maybe email me separately and we can discuss your involvement in the WIO2 project. Cool, thank you.
Great. Well, thanks for all your replies, Tanya. I guess that brings us to the end of this session and the first day of the workshop. So just very briefly to sort of summarize what we have been covering today. The first talk was by Denan Parker, and we have been introduced to key concepts, including study design, population, sampling unit, etc., as well as some key considerations relating to spatial, temporal, and ecological extent of your research question. Then the next talk was by Alison Koch, and then we've introduced on the different data layers that you have to do, that you have to use during MPA monitoring. The process of consulting with the different stakeholders and trying to understand the different sort of uses of an MPA, and also think carefully about the sort of method that suits best for your monitoring, and some considerations in data management. And then during this session, we have been introduced to sort of how to design a sampling grid using GIS data, and how to automate this process by using a modeling approach, as well as consider depositing your data in repositories. And then finally, this talk, we have been introduced by Tanya to the different sort of types, examples of monitoring programs around the Western Indian Ocean region. Just to sort of remind you that tomorrow, do join us again at the same time, 8:30 UTC +2, so that is South Africa time. Tomorrow's session is going to be about data collection using Benthic camera systems, 5 different lessons on that, including a training video. And before I close the call and say, have a nice rest of the day to everyone, maybe, is there anything that I might have missed from the conference organizers that you might want to add at this stage? Silence is usually a no in Zoom, so I'm guessing all has been covered. So I would like to thank everyone for joining today and see you soon. Bye.